Sir, good morning. Outside.
Hello. Hello.
Good morning, sir.
Yeah.
Hello. Good morning.
Yes, I think Dr. Somnath, Dr. M. Patel has joined, and Dr. Alekha Das is with us. So
Yes, sir.
so you can start the program. Good morning, Mr. Sudhir. How are you?
Good morning, sir. Good morning, all of you, sir.
I am audible.
Yes, sir.
Huh?
Yes, sir. Yes, sir.
Okay. वॉइस कम आ रहा है शायद.
Yeah, it's okay now, sir.
Yes, sir.
Yeah. Madam, shall we start? Sneha, madam, you can start.
Okay. A very warm welcome to all dignitaries, professionals from industry and academia, delegates, and my teaching fraternity to the two-day 17th Indo-US Virtual International Conference on theme Recent Advances and Global Trends in Pharmacognosy, partnered with my co-host, Mrs. E. Navya Prawala, Assistant Professor, Department of Pharmacology, feel honored to greet you on behalf of St. Paul's
ના થઈ ગયો બંધ કરી દે.
Madam, your voice is not coming.
Okay. Now, sir. Now, sir.
Ma'am,
Am
it's
I
okay.
audible?
Yeah.
Okay. I hope we'll have a great day with the feast of scientific sessions by esteemed speakers of international honor, followed by scientific presentations of enthusiastic participants. The growing healthcare needs of world has posed serious challenges that met new era of pharmacy as leading frontier in healthcare sector. The global domain of pharmacy is recent trend, which always met the demands of drug development and discovery process by creating new innovative ways. Research in pharmaceutical sciences has cluttered the drug development process in sophisticated way to serve the human race. With the emergent COVID pandemic, the threat imposed, the pharmaceutical industry with its frontline warriors, the pharmacists were successful in serving the society. As we engage with increasing pace of pandemic, there is a need to understand emerging trends and advances in pharmaceutical sciences. Here, the virtual platform of INDO-US conference, we now share the advancements by sharing our knowledge and also dissemination of revolution in pharmacy to the budding pharmacists. It's my privilege and honor to invite our respected chief guest, Professor Dr. Madhabai M. Patel, Vice Chancellor, Sabarmati University, Ahmedabad, Gujarat, former Vice Chancellor, Hemachandriya, North Gujarat University, Pathan, Gujarat, to chair the conference. Thank you, sir, for accepting the invitation, and it gives me immense pleasure to invite our Honorable Chairman, Sri K. Sudhir Garu, and request him to inaugurate the two-day Indo-US virtual conference session. I welcome all the guests to the inaugural session of two-day 17th Indo-US Conference hosted by St. Pauls College of Pharmacy. Welcome to the grand inauguration. Thank you, Chairman, sir. Now, it gives me immense pleasure to introduce respected Professor Dr. Kiran Mai, Vice Principal, St. Pauls College of Pharmacy, convener for today's Indo-US Conference, to introduce to the gathering. Dr. M. Kiran Mai, presently working as Professor and Vice Principal at St. Pauls College of Pharmacy, has graduated B.Pharm from C. Padmavati Mahila Vishwavidyalaya, Tirupati in 1999. Completed her M.Pharm from JSS College of Ooty in 2007, where she was awarded gold medal. Madam has received Doctor of Philosophy, PhD, awarded in Pharmaceutical Sciences from JNTU Hyderabad in 2014. Madam has expertise of teaching and research experience of over 18 plus years in various pharmacy colleges. Madam has experience of chairperson for the scientific sessions in OU. Madam has published over 50 international and national publications and over 55 international and national conference papers. Madam has citations over 500 Google Scholar citations and Scopus Index citations. Madam is proficient in guiding projects of UG and PG, and she's expertised in establishing well-equipped laboratories. Madam was invited as guest speaker in International Online Scientific Congress, Phytomed 2020, organized by Melbourne, Australia. Madam has published a book chapter entitled Fabrication and Functionalization of Other Inorganic Nanoparticles and Nanocomposites in Taylor Francis Publishers. Recently, Madam received Scientific Excellence Award 2021 by Academic Growth Foundation 2021. I would now request Dr. Kiran Mai Madam to give a welcome note to the gathering.
Thank you, Sneha. Am I audible?
Yes.
Welcome, ladies and gentlemen, to today's 17th Indo-US Conference, International Conference, hosted by St.Pauls College of Pharmacy in collaboration with APP. Honorable Chief Guest of today's inauguration, respected Vice Chancellor, Parmamati University, Gujarat, Professor Madhabai Patel Ji. Honorable Chairman, St.Pauls College of Pharmacy, Sri K. Sudhir. Secretary Raghav Reddy. And our Honorable Principal Chandrasekhar Garu, and honorable guest of honor of today's inauguration, President APP American International Branch, Associate Dean Department of Pharmacy Sciences, Creighton University, Omaha, USA, Dr. Rajiv Dahiya, Director, School of Pharmacy and inspiration behind this conference, Faculty of Medical Sciences, the University of West Indies. Today's invited speakers, Professor Somnath Ji, Dr. Shivkan Kanya Ji, respected chairpersons of the today's session, Professor S. Lata, Anna University, SC Professor Dinda Sir from Tirthekar Mahavir University, Professor Y. Padmavati from G. Polarity College of Pharmacy, Dr. Sunita Dahiya, University of Puerto Rico, Sam John, PR, USA, and respected Dr. Somnath De, Organizing Secretary of Conference, my dear colleagues, other hosting partners, and all the delegates present over here, heart welcome to you all on behalf of management, principal, and staff of St.Pauls College of Pharmacy. So coming to the theme of the conference, it's recent advances and global trends in pharmaceutical sciences. So I'm not going in deep into the subject topics because a lot of eminent people are waiting to deliver their lectures. So as a part of my duty, welcome remarks. So just I would like to mention top 7 trends in pharmacy to watch out for, which is in relevance with the theme of the conference. And they are pharmacy supply chain automation and artificial intelligence boosting advanced drug discovery, digitalization of pharmacy industry, striking shift in conduction of clinical trials, focus on beyond the pill healthcare, shifting towards patient-centric healthcare, revamping of manufacturing process, and lot many. These are the 7 top trending advancements in pharmacy in 2021 conclusion. This is now we are moving towards 2022 with a lot of hopes. Even pandemic is throwing challenges to the entire pharma sector. So we have proven ourselves a lot of times from last 2 years. And now also with the same spirits, we'll go ahead with all respects. We are grateful to all the supporters of the conference, including APP team, registration, scientific, and organizing committee, and especially student volunteers. We are fortunate enough to get supported by a number of pharmacy professionals to organize this conference and to convert this from blended mode to full online mode. And many of my professional colleagues accepted last minute invitations because of this change. I'm really thankful to them, and my heartfelt welcome to all of them to the conference. And coming to the hosting institute or institute, St.Pauls College of Pharmacy, it has been established in the year 2007. And courses offered are 4 years UG program, that is B Pharmacy. And proudly I am here announcing in front of all in this Zoom platform is we have accredited by National Board of Accreditation recently for our UG program with the support of all our stakeholders. Congratulations, St. Paul's. And we also offer PharmD 6 years course and PharmD PB 3 years course and MPharm 2 disciplines, pharmaceutics and pharmaceutical analysis. Our institution is notable as St. Paul's family. Why I'll explain you now. Now APP also has become our family. Raju Dahiya sir, thanks for joining us. And COVID-19 has been throwing challenges to the entire world in terms of waves. we all know that the talk of town is now third wave, right? So same with received challenges and ultimately accepted the challenge, not only acceptance, 3 times we have accepted the challenges in a different way. So during first wave, St. Paul's knocked out lockdown by conducting 10 webinars. So by obeying all the rules of the protocol of lockdown and online classes, so we could able to achieve 10 webinars and 1 week EFDP program international, that too, and 20 various social awareness programs. And during second wave, 2 webinars, 26 awareness programs, social awareness programs and outreach programs, and 3 quality initiatives at international level. And finally, successfully entered into the third wave. So because we can't stop it, so finally third wave is going on and proudly hosting a prestigious 2-day Indo-US conference in association with APP. So once again, wholehearted welcome to all the delegates across India and all eminent people over here, all the chief guests, guest of honor, and my management, principal, and all the staff, and the entire pharmacy professionals. So we all are looking forward for exotic, informative, scientific lectures, enthusiastic oral and poster presentations, and lot many interesting announcements. With this welcome note, I convey my sincere thanks to both St. Paul's team and APP organizers for giving me this opportunity. Welcome you all once again. Thank you.
Thank you, madam, for your enthusiastic welcome and throwing limelight on emerging trends in the pharmacy. I hope our guests and gathering feel the esteemed welcome by St. Paul's family. The Global Pharma Outreach through our virtual Indo-US conference hosted by St. Paul's College of Pharmacy is commendable. Now, I feel honored and it's my pleasure to introduce our chief guest for today's conference, Professor Dr. Madhabai M. Patel, to the gathering. Dr. Madhabai M. Patel, Provost, Sabarmati University, Ahmedabad, and is an Honorable Director for Sri Swaminarayan Sanskar Pharmacy College, Zindal. He is a former Vice Chancellor, Hemachandra North Gujarat University, Patan, Pharmaceutical Education and Research, Kalol. Sir has done B.Pharm and M.Pharm from LM College of Pharmacy, Gujarat University, Ahmedabad from 1975 to 1977 and got awarded PhD from Hemachandra North Gujarat University, Patan in April 2003. Sir has done LLB from Gujarat University in 1979. Sir has over 44 years of academic, including 20 years of research and 32 years of experience as a PG teacher. Sir, research interests include novel drug delivery systems, immediate and sustained release formulations. Sir has guided over 30 M.Pharm students and 58 PhD students. Sir has 251 national and international publications and 43 review publications, and he was a first prize winner for the papers at state level presentations for formulation optimization of modified release diltiazem HCl tablets using composite index. Sir has published 14 books and has guided 5 research projects. He is an ex-Senate member for Gujarat University and ex-member for Gujarat State Pharmacy Council. Sir is member and life member and also ex-chairman for many prestigious professional bodies. Sir has got Lifetime Achievement Prestigious Award by APP UP Bareilly. I gladly welcome our chief guest to the 17th Indo-US Conference hosted by St. Paul's College of Pharmacy. I would request our chief guest, Dr. M. Madhabai Patel, to address the gathering.
Thank you, madam, for introducing me.
Thank you.
I am audible.
Yes, sir. Yes.
Good morning to one and all. On the occasion of today's 17th Indo-US Virtual International Conference, organized by St. Pauls College of Pharmacy, Telangana, and I thanks to organizer. the Honorable Chairman of Trust and College, St.Pauls College of Pharmacy, Honorable Principal, respected Professor Kiran Mandwa, and Convener and Vice Principal of St.Pauls College of Pharmacy, Professor, the guest of honor, and President of APP American International Branch, also the Professor Muralidharan Rao, guest and honor on second day, the President APP Telangana State Branch, for on this conference that learned speaker Somnath Singh, Shiv Kanya, Gloria, and for the chairperson S. Lada. Especially I thanks, and it's my pleasure to say Dr. S.C. Dinda. I known to me well, I also known to him, and for organizing session 1, 2, 3, and 4 for 2 days. On this conference, the recent advances and global trends in pharmaceutical sciences, the topic is selected. Today inaugural in scientific session, Dr. Rajiv Dahiya, the president of APP, Sunita Dahiya, all the faculty members of St.Pauls College of Pharmacy, and respect dear students and all the members of management and faculty. It's my pleasure to say something on the day of opening the recent advance in a global trends in pharmaceutical science. It is a wide topic selected. I think today 7 lectures and second day also the learned speaker Sri Harsha, Lakshmi Prasanna, Sonu Beni, Sena Thakur, Dr. Dina V. Patel, Muhammad Sinha, and Monu Kumar Shukla. Really, they have spared a time on this occasion. On this title, the pharmaceutical sciences, it is wide topic, refers to, as generally said, it refers to category of scientific field, including discovery and design, drug delivery, drug action, clinical sciences, drug analysis, pharmacoeconomics, and regulatory pharmacochemistry, and also as a pharmaceutical sciences or the covers. According to the American Association of Pharmaceutical Sciences, they have covered number of disciplines that make up a pharmaceutical sciences. The multidisciplinary analysis and pharmacology, including quality assurance, biotechnology and research and development, clinical pharmacology and transfer research, drug discovery and development, formulation design and development, manufacturing sciences and emerging, including the medical devices, pharmacokinetics, pharmacodynamics, and drug metabolism, pharmacy and biopharmaceutics, regulatory science, ensuring quality and safety. In other words, some said that pharmaceutical science is the study of the formulation, development, manufacturing of new drug. New scientists have the power of transform patients' lives by developing a new treatment for the disease, especially at presently in COVID-19, the pandemic in condition that pharmacists have played role, and pharmaceutical science means as a biotechnology department, they are manufacturing the vaccines. And in the world, I think the India is the first had provided the maximum dose of the vaccines is prepared, and that is used for a specific for the disease, and it is given as social service in the that pandemic, COVID pandemic. In other words, pharmaceutical science is a field that focuses on the design and synthesis of potential drugs, as well as their evolution and biological system and disease models as a means of profiling their preclinical safety, efficacy, dynamic, and metabolism. The pharmaceutical scientist is a professional who perform a research and drive drug discovery, development, and testing. Generally, all we are doing that design, discovery, and finally that testing and after dosage form design. In a master also, pharmaceutics, pharmacology, pharmaceutical chemistry, pharmamedicine, and herbal drug. I think the branch of pharmaceutical sciences previously, before so many years, people are using for disease a herbal drug, Ayurvedic drug. They are collecting from the forest and they are using and giving the Vaidacharya Vaid. They are giving the treatment. After that, thought that the suppose on natural drug, it depend upon the rain on monsoon season. If the no rain is there, then how we will get the drug? Then they have scientists had synthesized same molecule in medicinal chemistry or pharmaceutical chemistry. But after synthesized number of molecules they have got, then how many they are useful or not useful, some toxic, non-toxic, then it should be required the clinical trial or pharmacological screening. After pharmacological screening, they found the molecule is very useful as pharmacological activity in a specific disease. Then after the pharmaceutics branch, they have tried to formulate in which way it should be marketed. Then as a pharmaceutics is the discipline of pharmacy that deal with the process of turning a new chemical entity or old drug into a medication to use safety and effectively by patient. It is also called a science of dosage form design. The many chemical with pharmacological properties but need to special measure to help them achieve a therapeutically relevant amount and their site of the action. That branch is pharmaceutical formulation, pharmaceutical manufacturing, dispensing pharmacy, pharmacotechnology, physical pharmacy, pharmaceutical jurisprudence, and clinical purpose of the drug depend on there. Actually, in any formulation to be required for the testing, I show you the one example in Gujarat. Before 20 years, they had formulated one that API. After that screening, it will be used for antihypertensive. Finally, it will be given for the clinical trial to the doctors, and the opinion of the doctors, they found that on 70% of these patients, that due to that particular drug, that hair growth on skin. After that, it will be not marketed because it is a side effect of this particular molecule, the effect of the antihypertensive. But their scientists had decided that if the side effect is hair growth on the skin, then they have a try to market for hair growth externally. And at that time, they have put in market the Grow More, the name, and 10 ml of this compound cost is in the market 110 rupees. Means it is testing and clinical trial is also required for any drugs. Pharmaceutical science, the advancement is presently in a wide field in a microsphere. Recently, some of example of their recent scenario being used to the drug delivery carrier is used as the carrier. Pharma innovation in most dry disease used with the heterocyclic derivatives in a several biological activities, and they found and that compound would be afterly it is marketed. The science in the first development in 1965, the liposomes and superior liposomal formulation over conventional delivery system, epidemiology, thalassemia in a generally inherited disease resulting in a descendant loss of hemoglobin and development. It is emerging as a global trend. In recent advance in traditional plant drug and at that it's other same that plants as a herbal drug or a medicinal plant, or we say the Ayurvedic drug. Especially we said that here the disease is osteoporosis, the frequent disorder having the brush familiar factor. We know that what is the effect of the osteoporosis, but finally if the drug is found. I think recently I read one article that other drug is the only the minimum weight tablets. It may be it is a less cost compared to layer tablet or tablet in tablet, and the lack of strategic drug solubility, stability, decrease in adverse effect. Recent development in nanotechnology for the treating a metastatic melanoma and emerging assist to reduce the extent of problems that other drugs therapeutics means that presently the work is going on nanotechnology. Same way trends in innovative pharmaceutical industry, it is a fight against a new existing threats, increasing of longevity. The complexity of drug, monoclonal antibody, the drugs, especially in anti-cancer, personalized medicine, treatment of the increasing aging population, arthritis, control release drug, three-phase tablets, the biopharmaceuticals and biogesic market potential drug product may be the biotechnology, the stem cell therapy, technology and automation, future pharmaceutical R&D workforce, stronger genetic markets, generic markets of the drug in future. One patient is of generic compliance, the generic equivalents. Two-factor gain the generic drugs, immediate access to the market, reduce the cost of consumer, and coordinate by the drug control authority of the safety, quality, and efficacy. The emerging new market, but more recently convert from the Asia, Latin America have emerged with the remarkable market growth in a pharmaceutical science in pharma industry. The innovative distinguish the trends in diabetic medicine. Trends in diabetic medicine in US 1984 to 2030. The strategic trends in pharma innovation. The new drug development phase, it especially required to more than 5 to 7 years. Pharma sector highest R&D, the scope is there. Pharma innovation, the challenges in recent times, new chemical entity research, new combination research, patient compliance, especially that look on the patient compliance, reducing the dosage frequency, improve the delivery devices, improving the across the potential and personalized medicine. And especially at a present, the natural products, tissue culture and produce activity, setting and QA and regulatory standard and randomized delivery means so many that challenges with so many innovation was there in the pharmaceutical sciences. I not say on this topic more because the London speaker, they had organized so many good lectures and they will speak for about on the our theme of the recent advances and global trends in pharmaceutical science. I conclude my talks. I thank to organizers, especially Rajiv Daya and principal of this St.Pauls College of Pharmacy. They have inviting me on giving a chance to speak few words over here. I especially thanks to Honorable Convener and Vice Principal of the college and management and faculty. I thanks again to organizer, and I congratulate for organizing this conference, and especially what they had said that college is also a NBA accredited. So I again congratulate for the accreditation, NBA accreditation, and for giving a quality of the education. I thanks to all. Thank you very much.
Thank you, Patel sir. Thank you so much for accepting our invitation. And thank you, Rajiv Dahiya sir, for inviting such a nice personality to our conference. Thank you, sir.
Thank you. Thank you very much.
Thank you so much, sir, for your valuable insights into each and every discipline of pharmacy and need of upgradation and advancements occurred along with the emerging trends in the pharmaceutical sciences. And now I'll also welcome Professor Sunita Dahiya, Secretary APP, Dr. Ajit Singh, CEO Climate Solutions, and Dr. Karthikeyan, HOD, Professor Majid Iqbal from ESCO College of Pharmacy, and Professor Y. Padmavati Madam from G. Polarity College of Pharmacy to the 17th Indo-US Conference, as they have joined lately. I now gladly welcome our guest of honor, Professor Aleka Dash, President APP American International Branch, Associate Dean Research, Department of Pharmaceutical Sciences, Creighton University, Omaha, USA, to chair the conference.
Good morning,
Thank you
all.
for accepting. Yes, sir.
Good morning. Hopefully, are you hearing me?
Yes,
Yeah,
sir.
good morning. We can hear you well, sir.
Okay, thank you. And good morning again to all the guests, honorable guests, delegates, speakers, as well as the students. This is my great honor to speak few words. I think I don't want to take too much time because the speakers are waiting and there are beautiful and wonderful and exciting topics that will be discussed. I think that is more important. But one thing I just, when I was opening the announcement of this particular 17th Indo-USA Virtual International Conference, and also the theme, I found one thing very interesting to me. It talks about in conjunction with Parakram Divas, which was in respecting one great soul of India, Dr. Netaji Subhash Chandra Bose. I don't know how many people have known that great soul. Actually, the reason I'm bringing it, who was born in 125 years ago on January 23rd and of 1897, was born in my own state, Odisha. He was born in Cuttack. I was not too far from that place. So that's why I just want to bring to the attention such a great soul. And we are dedicating all this particular seminar in the pharmaceutical sciences in his honor. That's a great things to start with. In regards to the topics, I don't want to spend too much time. People are talking about COVID challenges. As I have told in the past, whenever there is a challenge, it comes with an opportunity. And this has given opportunity for all of us, especially the pharmaceutical scientists of the world, to come up with some ideas and some innovation to tackle the challenge. And one of the token of that particular hard work has been already seen in practice. And as you all know, that these pharmaceutical scientists, with their hard work, could bring this vaccine so quickly and saved millions of people. Unfortunately, we also lost millions of people. But we are not the only people who can able to fix the problem of the world. So one of the things we learned over the years, that teamwork is essential for any success. And this is one of the lessons we should all learn that whether I'm a pharmaceutical scientist, whether my discipline is pharmaceutics or even medicinal chemistry, or it can be pharmacology or social administrative sciences, we have to work together as a team. So only the teamwork will be the success at the end. So with that, I don't want to talk about the different topics. I don't want to talk about what are the things will be discussed. Each and every topic, whatever small, whatever big it is, it has a particular purpose for a success. And one of the classic example I give it to my students when I teach, is think about a pizza, and it consists of so many things, starting from flour, starting from many different types of additions to make it more palatable and better. But a small pinch of salt, which is not required in that amount, but it's invaluable in that particular palatability, or in particular to make this pizza a better pizza of the world. So don't feel that your branch of pharmacy or your branch of science has nothing to do with the development or the problem solving. So with that things, I'll strongly encourage our students that they're our future pharmaceutical scientists of the world. So don't underestimate yourself. Work on your strength, and hopefully one day we'll be giving and we'll be proving that this profession of pharmacy will bring to the table and serving the humanity for many, many, many years to come. With that, thank you all for the invitation and wish you all great success for the next 2 days. Thank you again.
Thank you so much, sir, for enlightening us with his enthusiastic words. And now I take the privilege to invite Dr. Rajiv Dahiya, Director, School of Pharmacy, University of West Indies, St. Augustine, Trinidad Tobago, to chair the conference scientific session. Dr. Rajiv Dahiya has received. Okay, sir.
Madam, you can continue.
Thank you, sir, for
Madam, you can
accepting
continue.
the invitation.
Shall I continue?
Dr. Rajiv Dahiya, sir.
Hello, am I audible?
Yes, yes.
Yeah. So
Please system, please system.
I welcome all of you in this 2 days 17th Indo-US conference, and a hello from Trinidad and Tobago, University of West Indies here. First of all, I would like to thank the organizers, mainly chairman of the St.Pauls College of Pharmacy, as well as I would like to appreciate the efforts of the convener, co-pattern, and the organizing secretary for the last month or so for their hard efforts to come to a concluded event today. And as Professor Das said, we would like to salute the contribution of Netaji Subhash Chandra Bose. We would like to memorize his quotes, what he did for the freedom of the country, and we remember him very much on this very occasion. So, although all things were spoken by the earlier speaker, so I would like to highlight
Unstable.
this conference will be reached with the 5 scientific lecture sessions from Somnath Singh from Creighton University, Omaha, Dr. Shiv Floria from AIMST Bedong, Kerala, Malaysia, Professor Kuntal Das from Parmamati College of Pharmacy, Bangalore, Karnataka, Professor Satish Kumar from Wells DW University, Chennai, Tamil Nadu, Professor Sri Devi from Sri Padmavati Mahila Vishwavidyalaya, Pune, Andhra Pradesh. Besides this, there will be 7 orations from different states like Odisha, Andhra Pradesh, Kerala, Telangana, Gujarat, Tamil Nadu, and Himachal Pradesh. There are 7 speakers were included in the orations, which will be a competition for Outstanding Oration Award, and that will also be a interesting one for in terms of research. So, without taking not much time, I would invite the anchor to introduce about speakers and start the program. Thank you very much for giving me a chance to speak.
Okay. Then remove chair.
Thank you, sir. I would now like to invite our organizing secretary for Indo-US Conference 2022, Dr. Somnath Dev, Head Department of Pharmacology, to chair the session. I now call upon my partner co-host for today's event, Mrs. E. Navya Pravala, Assistant Professor, Department of Pharmacology, to take up the scientific session and invite speakers, chairpersons for the scientific sessions. Thank you, Dr. Sneha. I now take the privilege to introduce today's chairpersons for session 1, Professor S. Lata from Anna University, Tamil Nadu. Dr. Subaya Lata completed a bachelor's degree in pharmacy at Dr. MGR Medical University, Chennai, a master's degree in pharmacy, and a doctor of philosophy in pharmacy at Jadavpur University, Kolkata, India. Currently, ma'am is working as an associate professor in the Department of Pharmaceutical Technology at Anna University. She has 20 years of experience in higher education and research gained at Jadavpur University, Bharati Dasan University, and Anna University. Ma'am has broad knowledge in pharmaceutics, pharmaceutical engineering, and pharmacology. Her research interests include the development of novel drug carriers using magnetic nanoparticles for targeted drug delivery. Ma'am's lab has received competitive research funds of 75.52 lakhs rupees. She is a visiting scientist to Technisch University, Germany, Yokohama National University, Japan, and Shizuoka University, Japan, University of Ibadan, Nigeria. She is the author or co-author of over 70 research publications and 12 book chapters, and edited a book titled Nano-Biopharmaceutical Technology, Applications and Perspectives, published by Elsevier. She has supervised over 1 postdoctoral, 3 PhD, and 42 master's students. She has been the recipient of 2008 Tamil Nadu Young Scientist Fellowship for Young Scientists, the 2014 Tamil Nadu Young Women Scientist Award in Medical Sciences discipline in recognition of outstanding research in pharmacy. The 2014 late Srimati Shaima Dahiya Memorial Award for the Outstanding Pharmacy Teacher, and also 2018 Young Pharmacist Award from the Association of Pharmacy Professionals. She is a life member of many professional bodies.
Oh, yes, sir, please.
Yes, ma'am.
Yes.
I invite respected Professor S. Lata to chair the session. Thank you, ma'am, for accepting our invitation. I now invite respected Professor S.C. Dinda from Tirthankar Mahavir University, Uttar Pradesh, India. Professor Subhash Chandra Dinda has did his B Pharmacy from Perambur University, first class with gold medal, M Pharmacy from Andhra University, first class with branch topper, PhD from Jadavpur University, and honored with Life Fellow from Institute of Chemists, India. He is working as professor for the head of Pharmaceutics College of Pharmacy from Tirthankar Mahavir University, Moradabad, UP, India. He is a former professor and coordinator of pharmacy, Mekelle University, Ethiopia, and professor and director of pharmacy, Berhampur University, Berhampur, India. Sir is having 24 years of professional teaching, research experience, including 16 years of administrative experience as principal, director, coordinator, and dean of faculty of pharmacy in India and also in abroad in Ethiopia. Sir has research contributions like 3 patents and 6 books and also 123 articles in referred journals, which has index of 21 and I-10 index of 50, and he has total citations of 1408. He has supervised with 23 doctoral theses. To his credit, he served as vice president followed by president, Association of Pharmaceutical Teachers of India, Odisha State Branch from 2006 to 13. To his credit, he also served as Associate Secretary, Indian Association of Pharmaceutical Scientists and Technologists from 2009 to 15. He is a member of Academic Council, Biju Patnaik University of Technology, India from 2003 to 2007. He is also a member and chairman for Doctoral Research Committee, Board of Studies, Berhampur University, India from 2008 to 13. He is a member of Graduate Council of Pharmacy, Mekelle University, Ethiopia from 2014 to 18. He is Dean and Chairman, Board of Studies and member, Academic Council, Rama University, India from October 2018 to May 2019. He is also a member of professional bodies like CRC, IQAC, and Academic Council, Tirthankar Mahavir University since October 2019. He is also an editorial board member, reviewer of many journals, including Elsevier, Springer, and Science Direct publications. He is a recipient of Gold Medal for B Pharmacy in 1996, Priyadarshini Indira Gandhi Award 2005, Arc of Excellence Education Award 2005, ISTE Rajalakshmi Memorial Best Teacher Award for Orissa State 2006, Life Fellow of Institute of Chemists India 2008, APP Distinguished Teacher Award 2015, PDGOODS Outstanding Scientist Award 2019, and Shiksha Bharati Award 2021. I welcome you, sir. Thank you, sir, for your presence.
Thank you for introducing me. Thank you.
My pleasure, sir. Now I feel honored to introduce to the meeting our esteemed guest, Professor Somnath Singh from Triggerton University, Nebraska, USA. Dr. Somnath Singh is a professor of pharmaceutics at School of Pharmacy and Health Professions, Triggerton University, USA, where he has began teaching pharmaceutics, drugs, and pharmacogenomics in disease management since more than a decade. His teaching philosophy is based on problem solving and active learning, which involves various educational technologies for enhancing students' learning outcomes. He has been awarded 5 times Educator of the Year award by his students. He is also director of the graduate program in pharmaceutical sciences, where he is responsible for overall administration and student success. Recently, the program completed its self-study needed for programmatic evaluation and accreditation. His research incorporate analytical method development, controlled deliveries of therapeutic protein and peptide, strategy and biological activity. Permission enhanced dermal method for quantification of aqueous solution of hydrogen sulfide, sustained release delivery system for hydrogen sulfide donors, nanogel formulation of curcumin and for the prevention and treatment of melanoma. He is a member of American Association of Pharmaceutical Scientists, Controlled Release Society, American Association of College of Pharmacy, National Geographic Society, and National Parks Conservation Association. He has been invited to a member of Alpha chapter of Rho, Chi, and Pi Lambda Sigma, Alpha Mu chapter, which are the only honor societies at his school. He is an editorial board of International Journal of Pharmaceutical Analytical Actor and an external PhD thesis reviewer for Dr. APJ Abdul Kalam Technical University, Uttar Pradesh, and Birla Institute of Technology, Mysore, Jharkhand, India. He has written 2 textbooks, 3 book chapters, published 29 manuscripts in peer-reviewed journals, and presented 69 oral and poster original research presentations in various national and international annual conferences. Now I request Professor Somnath Singh to address the session to give insights on the topic, Genomic Revolution in Pharmacy Practice and Drug Development. Thank you, sir, for accepting our invitation. We welcome you, sir.
Thank you so much. Thank you so much, Ms. Thakur, for introducing me. I don't have ability to share my screen, so whoever is host, can you please enable me to share my screen? So that I can share my PowerPoint slides.
Professor Sinha, allow Professor Singh to share his slide.
Yes, sir.
Still, it is showing host disable participant screen share.
Yes, sir.
You can click on my name and you can make me co-host, then I will have ability.
Yes, sir.
Yes, sir. It has been completed, sir. Now
Yeah,
you are the host, sir. Yeah.
Yeah, thank you. Good morning, everyone. So today I'm going to speak on genomic revolution in pharmacy practice and drug development. I selected this topic because it is matching with the theme of this presentation or this meeting. And for that, I am thankful to APP Karnataka as well as American International Branch and St.Pauls College of Pharmacy to speak on this topic. So let us start with what is pharmacogenomics. So long story short, pharmacogenomics is the influence of our genome on pharmacokinetics and pharmacodynamics of drugs. There is another term and which is related, pharmacogenetics, but it is a little bit different. And the distinction is pharmacogenetics are mainly concerned with our the influence of our inherited gene. On the other hand, pharmacogenomics is changes in anywhere out of our 3 billion base pair, and what those changes can do on the drug efficacy. And the ultimate goal of pharmacogenomics would be individualized or personalized drug therapy. So the basic philosophy of pharmacogenomics is okay, right now what is happening? Okay, that same standard drug is approved and clinically used for each of us. But God has made each of us unique, different. Means that we differ in our 3 billion base pairs, and therefore that pharmacokinetic and pharmacodynamic effect of a drug would be different. And it happens that even Tylenol, like paracetamol, can cause some severe liver toxicity in some rare patient. Otherwise, it is safe for so many of us. So the benefit of pharmacogenomics is or are that the future medicine would be more powerful and more precise. For example, right now there are some development. There was a first drug in such area like Bidil, which is approved by US FDA only for African American population for angina, not for other ethnic group, because it does not work in other ethnic group. The recent COVID vaccine is another example that so far the fastest vaccine developed in the history. It took 7 or 8 years, but now everyone knows that Pfizer and Moderna vaccine took less than 1 year. And the reason was that these medicines are based on messenger RNA. So like before these Moderna and Pfizer vaccine, the basic philosophy or approach behind vaccine was that the disease-causing virus was injected, although a weaker strain, so that it will not cause full-blown symptoms of the disease. And therefore, in the classical vaccine, it was taking more time, and it was more risky. But now, on the messenger RNA-based vaccine, now we know that the antibody produced in our body, it needs which antigen, that is protein. As many of us may have heard, this COVID virus are having a spike protein which helps in attaching. So that spike proteins, if once inside our body, will activate immune and to produce antibody. So this messenger RNA is like RNA is responsible for the production of proteins. So they are highly specific that which one proteins out of all the proteins in spike proteins, that which part is most efficient for causing the production of antibody. So this is the benefit of pharmacogenomics. Its economic consequences would be that the drug development is highly expensive. For one drug molecule, generally it takes 10 billion. And the reason is why most of the drug gets unsuccessful in clinical trial, because each of us are different, and that's why the drug which was working good in vitro setup in most of the cases may not work in clinical trials. But by the use of pharmacogenomics, now we can pinpoint that which one drug can work in which kind of population having what a specific mutation or polymorphism. There is a recent example, not recent, maybe 2-3 years old, that that Keytruda, yeah, is a medicine. It works as a miracle in lung cancer, but it works only if the patient is having PDL1 mutation, not in everyone. So this is another examples of use of pharmacogenomics in the drug development. So here is a diagram and reflecting whatever I talked about so far, that in the center group, it shows that different kinds of human. So you can see there are different color. So right now, whatever the standard practice, that you can see on the top left corner that human colored by green, that the standard drug in a standard dose, it is toxic, but it is showing beneficial therapeutic effect. On the yellow colored, you can see that the drug is not toxic, but not therapeutically beneficial also. On the top right corner, you can see that the drug is toxic and not beneficial. In the lower left corner, you can show, you can see that only lower left corner is useful or ideal, that drug is not toxic and it is beneficial also. So by use of pharmacogenomics, we can minimize this problem. And this slide is showing that that right now the basic pathway of disease symptoms is what? That if our DNA is mutated, then RNA will also be translated abnormal, which will cause abnormal protein, and which ultimately leads to the clinical phenotypes, that is the disease symptoms. So the disease biomarker, right now biomarkers are mostly chemical based, but when the mutated gene or polymorphism will be used as biomarker, then our diagnostic progression of the disease, as well as severity of the disease, all these three areas will be more precise. Here are some interesting facts, that here are some kind of prediction that what will happen in 2030 and 2040 in the area of pharmacogenomics, or due to application of pharmacogenomics. So it is expected or predicted that by 2030, the human genes responsible for our aging will be fully cataloged. It means we can live maybe more than right now. But it is debatable whether living this much, extending human life is good or bad. It is, yeah, it is debatable. And there will be computer model for all the human body, all the human cells, and therefore the good thing that maybe we will not have to test any drug on live animal. So that's the good thing. And this forecast that complete human genome sequencing will cost less than 1,000. You may have heard that human genome was first sequenced around 2001 or 2002, our 3 billion base pairs, and at that time it took about $1 billion. $1 billion. Now the prediction is by 2030, 1,000, but this has been realized maybe 10 years ago about. And here is a FDA link, which is interesting because now FDA provides recommendation as well as some mandatory instruction also for a specific drug, which kind of genomic screening should be carried out before that drug can be prescribed. So you can see that future medicine, here this color diagram shows that future medicine will work equally good in all because it will be tailor-made. Now drug metabolism. So our genes or the mutation in our gene affects the metabolism of the drug, and this is highly significant because depending on whether the drug needs to get metabolized, if the drug being used as a prodrug, then it needs to get metabolized for the production of active drug. Or if a drug taken is already active, it is not a prodrug, then at what rate it is metabolized, slower or faster, it will have significance. So here are some data, and this data is talking about like CYP2D6. This stands for cytochrome P450 enzyme, or gene responsible for producing that enzyme. Cytochrome is involved in metabolism of so many drugs. That's why it is also called ubiquitous enzyme responsible for metabolism. So if a drug is metabolized by oxidation, this cytochrome P450 is involved. And there are 48 variants or mutation of cytochrome P450. Here is one, like one of the 48, cytochrome 2D6. And this mutated form of cytochrome has been investigated extensively. That's why we are talking about. So this mutation is found in different frequency in different human population. And here it is shown, for example, for Chinese or African and European. So we will see some of its application in our later slides. Here is some interesting historical story about pharmacogenomics, that isoniazid is a dirt cheap, very cheap drug for tuberculosis. Everyone can afford, but in TB, tuberculosis, it is used for years, like chronic use. So it was found that in some of the patient, it is causing toxicity. Toxicity was like a neurotoxicity. And due to that, the patient was like a hand was trembling, uncontrolled. So that's why the investigation started. Why some patient is having neurotoxicity due to use of isoniazid? What is the reason? So isoniazid is metabolized by enzyme N-acetyl transferase. And there are two variants of gene producing N-acetyltransferase enzyme, NAT1 and NAT2. And isoniazide is mainly metabolized by NAT2. And there are 15 variants of NAT2. 4 are rapid, 9 are slow, and 2 is still not figured out. So here is a specific example. If at base pair 341, if thymine is substituted by cytosine, then that is leading to slow acetylation. And here is some data that in white Caucasian population, about 45% of population is slow acetylator. So if you are slow, it means that isoniazide will keep on depositing and it will cause a toxicity. And here are some other drugs which are also metabolized by same enzymes. And here is interesting some fact that NAT1 or NAT2 enzymes are also involved in metabolizing environmental pollutants. So now some research is going on. Is there any relationship between cancer and whether you are slow or fast metabolizer for acetyltransferase, which kind of acetyltransferase who have. If someone is fast metabolizer, maybe that person can metabolize the environmental pollutants and may not get cancer due to that. So the research are going on finding that kind of relationship. The influence of metabolism, as I started with, is dependent on which kind of drug. If a drug is pro-drug, that is, the drug needs to get metabolized to work, then if a patient is a poor metabolizer, that is slow, PM, poor metabolizer, then drug efficacy would be 0% of the standard efficacy. Then you can see if it is normal metabolizer, yeah, it is about 80, 90 or 100%. And therefore, it is essential to figure out which kind of metabolizer who is. For cytochrome, there is a Roche company kit using which anyone can determine which kind of cytochrome out of 48. And it cost around $250. But once anyone figured out, and it is good for whole life. Now, we are talking about mutation or polymorphism. SNP stands for single nucleotide polymorphism. As the word indicates, that out of 3 billion base pair, even if there is a single change, it can have some consequences. So out of our 3 billion base pairs, about 3 to 5% are coding sequences. Coding means those base pairs are responsible for production of proteins. If proteins are abnormal, we will have disease. So if a single nucleotide polymorphism, or if a polymorphism exists inside coding sequences, that will have profound significance, because then it will interfere with normal production of protein. But the good thing is that coding sequence by nature are highly conserved. That is, less mutations are found. So most of the polymorphism are found outside of coding sequences. So, but it is not a matter of celebration, because even those sequences are important, even outside, because they can predispose for a particular disease. So you can see that someone is walking in cold, not having any warm clothes and no catch of cold, and someone is more susceptible. So SNP can predispose. Another interesting example of pharmacogenomics is in cancer. And the typical example here I am showing about thioguanine. Thioguanine is used in blood cancer. So for thioguanine, its a prodrug is azathioprine or 6-mercaptopurine. So in this diagram, you can see there is an enzyme TPMT, thiopurine methyltransferase enzyme. And so you can see this enzyme is working at the two step that it is converting 6-mercaptopurine to methyl mercaptopurine, or mercaptopurine nucleotide is being converted by same enzyme by methyl mercaptopurine nucleotide. And thioguanine is the active form of the azathioprine. Like there are reports or there are data that someone can be someone can have low TPMT, means that a thiopurine methyltransferase enzyme is metabolizing less. It means a standard dose of azathioprine would be a high dose. So it will cause a toxicity. If it is metabolizing, if the patient is having a highly potent form of TPMT, then it will metabolize extensively. It means a low dose, that is underdose, so therapeutic failure. And here are some data that in Caucasian, they are having low metabolizing efficacy due to TPMT. Variant 3A, Korean has 3C. And here is some interesting YouTube video, but I will not play it for the sake of time. I don't know how much time I spent, but I am afraid I will take more time. So there is FDA guidelines that if anyone is having a low metabolizing ability, then 1/15, that is 15 times less dose of azathioprine should be given to that patient. And there are tons of cases that in chemotherapy of cancer, so many patients died due to the treatment. So who knows, the reason may be a standard dose is high dose, that patient may be low metabolizing form of that particular enzyme. Here is another drug, and which is used as antidepressant, and many of us know that with antidepressant the greatest problem is that in so many patients it does not work. And that's why you will see that patient under treatment for antidepressant, there are so many incidents of like suicide. And the reason could be that the drug did not work. And therefore, I'm very much interested in talking about these two drugs. Both are tricyclic antidepressant drug, and both are metabolized by cytochrome 2D6. So if FDA guidelines is, if a patient is either ultra fast metabolizer or poor metabolizer, in both kind of patient, these drugs should not be given, because whatever the dose right now, it is for standard for everyone. So, so if a drug is metabolized less slowly, then maybe that patient is getting high dose. If it is ultra fast, then low dose. And therefore, the recommendation is that try another drug. If there is no alternative, if there is no other alternative, other drugs are not working, then the compromised recommendation is that in poor metabolizer, for example, amitriptyline, its dose should be reduced by 30 through 50%. If it is ultra fast metabolizer, then that person should get a more dose, higher dose, that is 1.4 times of the standard dose. If it is intermediate metabolizer, then instead of 30 through 50%, 25% of a standard dose should be given. So this is the FDA guidelines. It is not my recommendation, or it is not my statement. Here is one interesting case history. And again, here also the same enzyme, cytochrome 2D6, is responsible. So here the case is that there is one patient, SN, 21-year-old, he is Caucasian, and due to oral surgery, he is having pain. So the doctor prescribed him codeine phosphate. And codeine phosphate is a kind of prodrug of morphine. It needs to be metabolized to morphine, and morphine will reduce the pain. But this person is having cytochrome 2D6, which is a slow metabolizer. And that's why that the next day patient came with his dad, that there was no reduction in pain. Then the prescribing physician increased the dose from 1 tablet to 2 tablets. And even after increase in dose, there is no decrease in pain, and the reason is cytochrome 2D6. That is the slow metabolizer. Another is warfarin. Warfarin is a blood thinner medicine, and the patient under warfarin needs the strict management of warfarin, because if dose is high, then patient may die due to internal bleeding if blood is too thin. So here is one case in which the patient prescribed warfarin is having two kinds of mutation. This person is having cytochrome 2C9. This is another, as I started with cytochrome, there are 48 polymorphism, 2C9 is one. And this patient is also having mutation for the enzyme VKOR, and that VKOR is vitamin K reductase enzyme. Vitamin K is just opposite of warfarin. Vitamin K helps in clotting of the blood. Warfarin is non-clotting, blood thinner. So we will see that that how warfarin works, that it inhibits the vitamin K reductase enzyme, which is responsible for converting epoxide form of vitamin K to the reduced form. And reduced form is the active form. That is, reduced form of the vitamin K will help in the blood clotting. So warfarin inhibits this enzyme. It means now the active form of the vitamin K will not be formed. That is, blood clotting will not happen, and therefore, and thereby thereby warfarin helps in non-coagulation or in the blood thinning, and it helps. But this patient is also having cytochrome 2C9 mutation. Like you can see, a star 2, it means that all of us are having each chromosome 2. So like both genes are mutated. So this cytochrome, this form of cytochrome metabolizes warfarin. So due to the mutation, now warfarin will be metabolized less. So here the clinical significance is if presence of cytochrome 2C9 or VKO or C1 had not been detected, then this patient must have got a standard dose, and a standard dose must have been resulted into extremely thinning of the blood, and this patient might have died due to internal bleeding. So see the significance of pharmacogenomics. Another example is clopidogrel drug. So here also the patient for which clopidogrel is prescribed, this patient is having a mutation cytochrome 2C19 2'2', and due to which a patient is poor metabolizer. And so due to this poor metabolizer, so a standard dose of the clopidogrel is 75 milligram per dose. So for example, question to think about is based on patient genomic, MS is hypothetical patient, genomic information, would a standard dose 75 milligram per dose is sufficient? But clopidogrel is a prodrug, and it requires to be metabolized to produce the active form of the drug. But due to this mutation, cytochrome 2C19, patient is poor metabolizer, and therefore a standard dose would be a low dose. So the question is, should the dose be double? Question is, will double dose will work? Answer is no, even double dose will not work, because you can see that this patient both genes are mutated. So even if a dose will be increased, this patient is such a poor metabolizer that this will not work, and the recommendation is that some another drug should be prescribed for preventing the patient platelet to form a plaque, and which is more dangerous. Clopidogrel is antiplatelet drug. And again, here is FDA link. So by clicking on this link, we can see there are dozens of drugs for which there is FDA requirement before prescribing this medicine, for which kind of polymorphism patient should be screened. Only then patient should be prescribed. Now there are some ethical interesting significance. So according to American College of Medical Genetics, that there are 56 genes which are pathogenic. Means if there is any mutation in those 56 genes, the disease will happen. Now ACMG recommends or requires that if some genomic testing was done, so genomic testing, in addition to whatever gene you are asking for, it also reports other gene also if it is present. So now the requirement is, if any of the 56 pathogenic genes was discovered even incidentally, that is the testing was for something else, but this was also discovered, then it is a requirement that patient must be informed that you may have this disease because this has been found in you. Similarly, there is a requirement that if any of the 56 genes are suspected in a child, for example, less than 7 years old, because their parents are already having, then even that kid should be counseled that why we are collecting saliva or a blood sample and what does it mean. There is an interesting Supreme Court case in my state, Nebraska. I live in Nebraska. Nebraska is a state in USA. So the case was that there was one convicted patient, sorry, not patient, person, and he was convicted to 40 through 60 years in prison, and because he admitted strangling his girlfriend. But now the interesting pharmacogenomics aspect is that person was under treatment with sertraline. Sertraline, another antidepressant drug, and on genomic testing, it was found that patient was intermediate metabolizer. It means that person was getting a low dose of the drug, that is therapeutic failure. So that convicted felon went to court and requested to hear again because it is not my fault. And this news is published in a journal, and title was Pharmacogenomics Made Me Do It. That it is not my fault, that I am intermediate metabolizer, so I was not getting the required dose, so the drug did not work. So my chronic depression was not treated. And you can see that the Supreme Court, state Supreme Court judge agreed, but that person lose the case, and the only reason was that at that time in Nebraska there was no law pertaining to using pharmacogenomics information. Otherwise, that person could have win the case. Another interesting fact is that there is a 24-year-old patient, and that person is being tested for breast cancer gene BRCA1, because her mother already has breast cancer. So that 24-year-old was recommended to test for breast cancer causing genes. But now the implication is that person is under stress whether I should go for this test or not, because if I am found positive, maybe I may not get health insurance, or my health insurance premium may be high, or maybe that my employer can fire me because my employer will have to spend more on my treatment. So what should be done? These are the ethical questions. So in USA, in 2008, one law was passed, GINA. It was the last law passed by President Bush before Obama. So GINA is a Genomic Information Non-Discrimination Act. So that patient should be told, no, there is a law, and according to that law, any employer or any health insurance company cannot discriminate. That is, cannot charge different health insurance premium from you. But here is the catch. Catch is that in GINA, the loopholes or caveat is if employer is employing less than 15 employees, then although there will not be any different or increase in health insurance premium, but life insurance or long-term care insurance, that premium is not covered by GINA. So maybe we need to update this law. And then there are some interesting cases here also. For example, that if I am a healthcare provider, either pharmacist or physician, and a patient come to me, and I know this patient is having this mutation, which may cause this kind of disease. So the question is, should I inform close relative of my patient? Because you are close blood relative, you also may have that mutation, and there is a probability of in future getting that disease. So the question is a physician or pharmacist is responsible for informing the blood relative? And the court verdict is yes. Yeah, so that is also interesting ethical perspective. And here are some guidelines that healthcare professionals are liable for informing the blood relatives and so on. So for the sake of time, I will not spend much time on this. So I am thankful to all my students who enrolled in my pharmacogenomics elective course. It is elective, and that's why I'm thankful they enrolled, and they challenged me to learn every year some new thing. And I'm thankful to my school, which provided me opportunity to offer a course, and my department chair, Dr. Alekha Das, who always supported me whatever I try to teach and whatever I try to research. So thank you so much, and please forgive me if I went beyond 45 minutes. Thank you so much.
Thank you, Professor Somnath Singh sir, for a very elaborative and very, very informative presentation, particularly on the pharmacogenomics in drug designing and development, and particularly optimizing the drug therapy. So we are very much thankful to you, sir. I think it's a very current research topic of current research interest, and our all the participants are going to learn so many things out of this topic, and how this genomic sequencing and all going to be utilized for the therapeutic measures, or to get a molecule to give a therapeutic benefit or therapeutic alternates such as vaccine development and all, because particularly after pandemic, so how this gene sequencing, this mRNA technology, the DNA technology we are utilizing for development of vaccines, and particularly mutation cases, and how the changes going on throughout the world, and how pharmacists take responsibility in developing the therapeutic alternatives or therapeutic measures for combating all these diseases, and to control pandemics and all. Very, very elaborative presentation by Professor Somnath Singh sir. And I hope we widely covered. Now it is a question and answer session. I want to ask one question, sir, particularly what you have given, citing some very good examples of particular drugs and the gene sequencing and what alternating the therapy, how to optimize and all. Sir, can you tell what are the factors to be taken into consideration while optimizing the genetic materials or genetic engineering drugs, particularly in the research, while optimizing the therapy, as you have cited some of the enzymes, enzymatic reactions held responsible for, and how to control it and all like that. There are several factors. If you elaborate a little bit, so I think all the participants can get maximum benefit out of it. Those who are particularly involved in research, it will be more helpful. Thank you, sir.
Yeah, so yeah, thanks for asking this question. Beautiful question, good question. If there is a new drug in clinical trial, so now it is FDA requirement that the human subject recruited for a clinical trial must come from a different ethnic group. And the reason behind different ethnic group that they will differ, like there is a probability they will differ in polymorphism or mutation, so that it can be figured out how this drug is working, in which that is whether this drug can be approved for entire human or not. So as I told example of BIDIL, BIDIL, and there is a separate website also, bidil.com. So in a clinical trial, it was found that it is working great in African American. So the requirement is ethnic group, because at one time determining all 3 billion base pairs was kind of so much expensive that it was impossible to use in clinical practice or in clinical trial. That if for example 2000 humans are recruited in clinical trials, so determining 3 billion base pairs of each human was impossible, so much expensive, and therefore the recommendation was ethnic group. But as I told, now due to development in technology, it is becoming less and less expensive. So once a drug is found that whether it is a prodrug or that is how it is metabolized, then the only recommendation is the adjustment of dose, that is whether low dose or high dose. So FDA has that requirement. If dose adjustment is not working, then the recommendation is in that patient find alternative drug. This drug is not good for that kind of or the patient having that mutated form of the gene. So that is the significance. So that we can say optimization, that we are preventing. As I mentioned, like warfarin, that patient could have died if those mutations could not have been screened before starting warfarin therapy. So these are just 1-2 examples, but there are tons of such examples. So that kind of optimization we can do. So this is good for our profession. It means now the role of pharmacist in explaining the genomic test to patient, or now you can see that now different dose, it means that pharmacist will have to compound, because the pharmaceutical industry cannot make maybe 5 form of the same drug in automatic machine, which is making like thousands of tablet or capsules per minute. That is not possible. So like when individualized or personalized medicine would be much more standard clinical practice, there would be huge opportunity for the pharmacists. And India being a diverse population, 1.3 or 4 billion, so India has a great potential for clinical trials of drugs. And this is true. So many American company for clinical trial are going to India, and that's the reason, diverse population. So more diverse population you recruited for clinical trial, it would be easier for getting FDA or any regulatory body approval. So that's my 2 cents. Did I answer your question?
Sir, as you said, this one of the variation of the impact happens because of the race or say ethnic groups like that. It may be also that because of genetic this gender variation, or maybe the age, or maybe also we know because of blood groupings, also individual constitutional makeup or blood components differ. So I think these are all factors may sometimes interfere with the effectiveness of the particularly genetically engineered drugs or say vaccine products and all. Even though you know, because what I want to like cytochrome P450 or say enzyme of the liver, like that, I want to ask you some more constituent, particular constituent may impact on those aspects. So those who are doing research, they may take care, sir. If any component, like you told some of the enzymes, particular enzymes, while citing some examples, like that, if you know some of the factors which are taken to be very important factors, 1, 2, if.
Yeah, so we cannot change, for example, if we have cytochrome 2D6, so we are slow metabolizer for so many drugs, so we cannot change it. We cannot change our mutated or bad genes. This is maybe facts right now, but in future it may not be true. You may have heard the CRISPR technology. So like theoretically or philosophically, by using CRISPR technology, we can cut the bad genes and we can replace with normal genes. We can cut the mutated gene, but maybe right now, due to so many reasons, it is not approved. But you can see that in sickle cell anemia, that there is one interesting case, that like both parents were having gene due to which it was confirmed that all their kid will have sickle cell anemia. So at the embryo level, that gene, by using CRISPR technology, was cut and replaced with normal, and then embryo was again implanted, and when baby born, its genome was sequenced to figure out whether this person is having normal or mutated gene, and which was almost certain, but that person is not having mutated gene. So that's the potential of sickle cell anemia. Another
manipulation, genetic manipulation is also a success.
Yeah,
I think.
Or maybe 2-3 years ago, there was a great world news that one Chinese scientist unethically, means without getting any approval, and he used the CRISPR technology in another human baby, which was having that genetic disorders, and it was successful. It was successful, but it was unethical because to do this, you need like approval. Even for animal experiment, we need animal committee approval, right? And therefore it was a big news. So other than CRISPR, there is another maybe revolution. Next revolution after genomics is epigenomics, which I did not talk about. And since you talked the environment or food or life habits, or with age, maybe our ability to metabolize may change, all are correct. So all are related with epigenomics. So there are one interesting cases, and sorry if I'm taking more time. That,
time also shortage. I think others are waiting where to stop here. And I think
so this is correct. Whatever you mentioned, food habit, that they can influence, even if we have a bad gene or mutated, it may not get turned on. So turning on or turning off the genes is another huge area. So it will take time to talk about that.
So
and that's why our lifestyle matters.
Questions, sir, I am requesting all the participants, if they are having questions, they can put in the chat box, and Professor Singh can clarify them later also through email also, because time is very short. I think Dr. Daya sir, you can tell if we should conclude this, or if any question, 1-2 question asked by the public.
And we can carry, Professor can answer in the chat box if any question comes, and we can carry, because we are already 10,
yeah, we are already late. So thank you, Professor Somnath Singh sir, for nice presentation and information, and clarifying these question answers. Thank you, sir. Thank you for your sharing valuable time with us.
Thank you so much, and thank you for Dr. Dahiya, APP Karnataka International, and St.Pauls for giving me this opportunity. Thank you so much. Bye-bye.
Thank you, Professor Somnath.
Thank you very much, sir, for your marvelous speech on pharmacogenomics, drug metabolism, and genomics revolution on drug development. Also, a special thanks to you, sir, for patiently delivering speech at very odd time from your location with your valuable insights into revolutionary drug development process. There are no more words, sir, for your valuable contribution in making the session graceful. Now I humbly welcome Professor Y. Parmamati from Polarity College of Pharmacy, Hyderabad. Ma'am has done her PG diploma course in chemical analysis and quality assurance from University of Hyderabad. She did her M.Pharmacy from Andhra University and PhD from Usmania University. Ma'am has teaching experience of 26 years. Ma'am is in expertise in handling of instruments like LC-MS, HPLC, GC, FTIR, and UV-VIS spectrophotometer. Ma'am has guided 50 M.Pharmacy students in their project. Ma'am is a life member of professional bodies like APTI and IPA. Ma'am has got total 33 national publications and 20 international publications. Ma'am has chaired scientific lectures in various conferences. Now I request Professor Y. Padmavati to chair the scientific session. We welcome you, ma'am.
Thank you, Sneha, for your nice introduction. It's a pleasure to chair the session of Dr. Shivkanya. Thank you.
Thank you, ma'am. It's our pleasure. I also welcome Professor D. Sunita Dahiya from University of Puerto Rico, USA. Ma'am serves as Assistant Professor of Industrial Pharmacy at School of Pharmacy, University of Puerto Rico, USA. She completed her PhD from Gautam Buddha Technical University, Uttar Pradesh. M Pharmacy from Rajiv Gandhi University of Health Sciences, Karnataka, and B Pharmacy from AR College of Pharmacy, Gujarat. Ma'am has 20 years of experience in teaching and research. She has published more than 40 articles in peer-reviewed journals and authored several book chapters with reputed publishers such as Springer, Taylor Francis, and Elsevier. She is a reviewer in international journals and editor-in-chief of Scientific News magazine published by Association of Pharmacy Professionals. Ma'am is a recipient of grants of $30,000 from National Institute on Minority Health and Health Disparities from the National Institute of Health and $70,000 from Puerto Rico Science Trust for the research project on nanocrystals. Ma'am serves as General Secretary of Association of Pharmacy Professionals since 2011 and is life member of various national professional bodies including APP, IPA, APTI, IPGA, IHPA, and international bodies such as American Association of Pharmaceutical Scientists and American Association of Colleges of Pharmacy. Ma'am's research area includes formulation science utilizing complexation, solid dispersion, and nanotechnological approaches for the dissolution and bioavailability enhancement of poorly water-soluble drugs and controlled release drug delivery systems. Ma'am academic interest includes implementation of active learning methodologies such as problem-based learning, classroom seminar and journal club, small group workshops, students portfolio, and rubrics creation for assessment. Now I request Professor D. Sunita Dahia from University of Puerto Rico, USA to chair the scientific session. We welcome you, ma'am.
Thank you.
Thank you, ma'am. Now I gladly invite Dr. Shivakanya Fuloria from MS University, Malaysia.
Thank you, ma'am.
Good morning, ma'am. Ma'am has done her PhD in pharmacy from Amaravati University, India, M Pharmacy in the Department Pharmacognosy from Department of Pharmaceutical Sciences, Nagpur University, B Pharmacy from Amaravati University, India, D Pharmacy from Mumbai Technical Board, India, and also PDCTM from Noida, Uttar Pradesh, India. Her research interest would be drug design, synthesis, biological evolution, cell line studies for antibiotics, anti-tubercular, anti-inflammatory, anti-dengue, anti-diabetic, anti-HIV, anti-gingivitis, and anti-cancer agents. Another research interests are phytochemical extraction and isolation and in vitro pharmacological investigation of pharmaceuticals, medicinal plants, and other biologicals. Analytical method development and validation for pharmaceuticals, and also nano formulation and molecular characterization of synthetic and phytoisolates. Ma'am has professional experience of 17 years and 2 months as an associate professor at Pharmaceutical Chemistry Unit of Faculty of Pharmacy, Amherst University, Malaysia. Ma'am's publications are Fundamentals of Pharmacogenomics, Economics in 2019, Principles and Practices of Traditional, Complementary and Alternative Medicine in 2017, Principles and Practices of Industrial Management in 2015, Spectroscopy, Fundamentals and Data Interpretation in 2013. Ma'am has filed patents on composition for the treatment of urolithiasis. Ma'am's MOOC courses published are Basics of 1HNN-MR and Data Interpretation in 2017 and Udemy.com, Principles and Practices of Production Management in 2015 at openlearning.com, Introduction to Glycosides and Resins in 2015 at openlearning.com. Ma'am has received research products and grants like FRGS Grant Malaysia titled Establishment of TOP1 and EGFR Inhibition Mechanism of in silico dot and synthesis novel CPT11 analogs in oral squamous cell carcinoma in October 2020 for 3 years. She also received FRGS Grant Malaysia titled Discovery of Novel Benzodiazepine Analogs Based Silver Nano Formulation for Treatment of Periodontitis due to gingivitis, a study that encompasses molecular docking, 3D QSAR analysis, synthesis and silver nano formulation of benzodiazepine accompanied by toxicity in October 2018 for 3 years. Ma'am has researched, papers published are 72. Ma'am has attended conferences, paper presented are 65. Ma'am has research project supervision, ongoing B Pharmacy, 3 students, MSc 2 students, PhD 6 students. Ma'am is also a life member of many professional bodies. We welcome you, ma'am, and I request Dr. Sukanya Floria, ma'am, to take up the session on the theme technology-based citation and referencing tools.
Thank you so much, Navya ma'am, and thank you first of all to APP and Dr. Daya sir, madam, and principal of St.Pauls College of Pharmacy for giving me an opportunity to share my knowledge, because it's a platform where we are exchanging our knowledge to each other. So just a small briefing I'll give here. Can you see my screen?
Yes, yes, yes. It's visible. Yes, Shukanya ji, please carry on.
Okay,
Yeah,
thank
it's
you, ma'am.
visible. Yeah.
This is today's topic which I am going to discuss here, that is technology-based citation and referencing tools. So I think everyone knows how the referencing is important in case of thesis and manuscript writing. As a student, as a researcher, or as a guide, you have to supposed to know these all points about the referencing. So referencing is a scientific method which explains the data source by providing standard sets of information. So how it can helpful? It helps us to identify correct references, and it helps us to easily search any kind of the material which is relevant to our project or our research, or if you are giving any assignment to students, that referencing is one of the important tool we have to look into, and retrieval is possible with the help of referencing tools. So why we should include the references in manuscript or in thesis or everywhere? What is the importance of this referencing and citation? So first thing, it help us to give the credit to the ideas and works of the other authors, because normally we are fortunate now. The Google uncle is there, who is helping us to get all the information throughout globe, and we can refer many papers, research articles, and somewhere we have to give the credit to the author and ideas, because it's the research is I can say the pool of people working together and exchanging their knowledge. Okay, so it also allows other to find the original source, because if you remember, many pharmacological methods, these are developed by 70s, and these are right now also we are using. So somewhere we are quoting those original sources also, and it gives work information. Okay, someone is in right now, sir is in West Indies, madam is in USA, and you are in India. We want to work out something, so we can exchange our work with if we can get the information from this by referencing. And recognition and authentication of work, that is one of the important aspect of referencing. And citation, actually it is nothing but the act of quoting, and what it helps to help us to write from another, usually with credit acknowledgement. If you want if you want to give the citation, it's if you're putting numbering, maybe it's a Harvard style or whatever style, but if you are giving the credit to the person, to the idea, to the author. So what is the importance of citation? So here you can see the citation also help us to find out the source that gives the information from the external source where from the original article is there, and it helps us to put work into together, like maybe some are working continuously on one particular for example, in my university, many of researchers, they are working on a vaccine from last 25 years. So they have a number of publications, so it's linking to each other, and we can refer that work and understand easily. We can understand the work and justify the inferences, because when we see research paper, we come across the results and discussion and conclusion. Based upon that, we can get an idea what would be the outcome for this particular research, and it will help also to evaluate the uniqueness and scope of the manuscript. And finally, the main concept, acknowledge the work and conceptualize the research findings. So currently, actually, you might be aware, not as many students I can see here, there are different types of the styles by which you can give the numbering. One is Vancouver style, another is APA style, Harvard style, Chicago style, and IMA style. So actually these are the different styles, but basically as a pharmacy professional, I think most of us come across the Vancouver style and Harvard style. Normally, whichever paper or whatever we are referring, or maybe as a part of our day-to-day curriculum, and so when we give assignments, we ask a student to refer, either they can go for Harvard style or Vancouver style. And we have a MLS style also. These all are used in other field like literature fields where they need to write. So whatever style is there, the aim is only one. The referencing style may differ in their representation, but the information they will provide same. So whatever you're either you write in Vancouver style or Harvard style or whatever style, but it will provide the same information, and it will help. It will solve the purpose, the same purpose, because anyhow, whatever the ultimate aim of any references or writing, whatever way you write, the main aim is to get the information, the correct information, and to utilize that information in our literature, in our research, or in other aspect of the review article and other things. So as I mentioned already, there are different ways of writing the including the referencing style. So, but these referencing styles are based upon citation sequence. Sometimes you use the citation in number form, and it will appear in text in number form only. Okay. At the end also you will write in a number form only. And other style is citation name, like Mendy Harvard style. When you whenever you write, sorry, in Vancouver style, sometimes you give the numbering, and sometimes when you see at the end of your back of the thesis or any manuscript, you will see it will appear in alphabetical manner. And third one is name and year, where Harvard style come into picture, where you write the name and year, and here also you will include your references in alphabetical styles. So you can see different styles are there: MLA, Harvard, and there are different sources by which you can get the information, print and electronic books, then journals, electronic journals. Nowadays, repositories available online. You can get information from repository, web pages, then conference papers and newspapers. Many more like white papers are there, government reports are there, and thesis and unpublished work. So you can retrieve information from this. Then bibliography and referencing. So you can see here that these are the two things which based upon the Venn diagram you can see here, the bibliography includes the citation and which might not be included in your text, and these citations are included for further readings. While in your references, you have to give the particular citation. You need to cite the particular sources, and bibliography or references, it will appear at the end of your thesis, book, or whatever the manuscript you are writing. And the small video I would like to show you here. Sorry, maybe I'll share the sound with you.
Sources that you use, right when you use them. Not only does this show that you're an honest and ethical student, but it also tells the reader exactly where you found that awesome piece of information. So, how do you give credit to other sources in your assignments? You use in-text citations, which can also be called parenthetical citations. An in-text citation connects information you're borrowing from a source to a citation in your bibliography, which helps teachers and classmates understand where the information originally came from. Include an in-text citation in your paper when you use a quote word for word from a source, or when you borrow an idea or phrase from someone else. In-text citations have to be written in a special way. They require 4 things in this order: an open parentheses, the author's last name, the page number that you found the information on, and a close parentheses. In-text citations are placed inside the sentence, so make sure that the period is outside of the parentheses. If you are using a source word for word in your paper, you need to put quotation marks around it. This is called a direct quote. Let's say that I found a sentence on page 20 of The Giver by Lois Lowry that explains an important part of the story. If I take all or even just part of the sentence word for word and put it in my paper, I would need to include quotations around it and add an in-text citation, like this. Let's say you want to explain something that happened in The Giver, but you explain it in your own words instead of taking it straight out of the book. This is called a paraphrase, and while you don't need to include quotation marks, you still need to include an in-text citation. This will give credit to Lois Lowry's writing and let your teacher or classmates know where the event happened in the book. Remember, make sure that the first word in the in-text citation matches the first word in the citation of your bibliography. That way, when people want to learn more about the source in your paper, all they have to do is look for the same word in the bibliography to find it. Let's review. The in-text citation takes readers to a specific citation in your bibliography, and the specific citation tells the readers where they can find the piece of information you used. There are special...
So this is about the in-text citation. So this is one of the example you can see here in citations. So you can see on screen, and this is the Vancouver style citations. You can see here number style citations. So as this topic is, today's topic is a technology-based pre-referencing management system. So here there are a few online softwares which are available, which can help you to improve your manuscript writing, even in thesis also, because normally everyone's as a researcher or as a student also, we want something that can help us to write the references, because manually if you're writing references, it will take another, I think, 15 days to, if we are having 300 to 400 references and how to arrange them, maybe it will take another 2 to 3 months. So here the technology come into existence. So you can see there are the technology assisted pre-reference management. One important is first that is Zotero. Then you can use CiteFast, you can use EasyBibe, you can use even Microsoft Word. Okay, normally we want to use Microsoft Word for the references purpose, but there is one option which can help us to include the references in our text that I'll show you later. And then AutoBibe is there, Dossier is there, Citation is there, Bibliography is there, PubMed is there, and Google Scholar. PubMed and Google Scholar, if like you don't want to like include your software or don't want to download, you can take the help of PubMed and Google Scholar to get the references, to get the citations. So that I'll discuss in later part. So technology assisted paid reference management, you can see here EndNote is there, reference work is there, Cite This For Me is there. EndNote and Zotero almost equal. So I would suggest, as a if you're a student, maybe you're a researcher or maybe master student, if you want to use, you can go for the Zotero as one of the good tool for writing your thesis or writing manuscript. So whatever technology you're using, it helps us to support in retaining uniformity of reference style and reduce the risk of overlapping citations. So sometimes if you're doing manually, there might be chances you may use the same references 4 or 5 times while quoting everywhere. So there might be chances some references you overlooked and overlapping happen. So if you use the technology based the citation tools, it can reduce this problem and overlapping can be reduced. So the first which I'm going to discuss, this is just online available. It is a free tool actually, but because of it's a free, you may find many advertisement around this page, and this is a site fast. Okay, you have to go to the Google, you have to ask to the Google uncle and it will show you the site name, that is www.sitefirst.com, and this is the web link. You can go, and once you go, either you can go for the login, or maybe you don't want to log in, but I would suggest you go for login. It will retain your information forever. If you don't log in, then maybe for 3 days it will retrieve all the references, and after 3 days all references will disappear. So this is the way to how to create the account. Simple. You need the email ID and the normal usual process which we do normally to create the account, and then after you will this kind of the web page will appear on screen, and so you can see. As I told, you may create your account, you can log in, and once you log in, you can see there are different style. APA style is there. APA style is almost the Harvard style, and APA style is similar. So it will help us to get the references in a correct way. So there are different modes. You can see here, like from web page, you can get information. From book, you can get information. And there are other medias also. You can see from web page, government publication, audio, interview, all from that you can retrieve the references source. We can get it from using this particular site. Then after that, if you are referring to web page, maybe manually you can add on, or maybe you can just include the keywords, and you will get the information. Like for example, just now on screen, you can see just I type in, and I got the information and when it was published and how it was published. So the final step you have to do, you have to save the citation. So once you save the citations, so you can see here, you can get the references. Okay, so this is the reference which I retrieved from the web page. And then after this, you can see the index citations also, it is automatically giving, like how this is actually if you don't want to use any software or you're having trouble, so at least nothing is better, something is better than nothing. So this is like those who are having problem, they can use assignment. Students mostly they are preparing assignments, so where you need to code this particular things, presentations, so where you can use this particular tool. Then if you want to retrieve the references from book, you can use this particular step. Just type in the, as you can see, enter the book title or author name. So you can see just I included the author name here, and you can find whatever books we have been published that is appeared onto the screen. Okay, so just click on the book which I want to use as a reference. Maybe you can manually also do, and you can just type in, and you can set the citations, and the final stage you can see here the book references also appeared, and in-text citation also appeared here. Then after this, if you want to refer the journal, maybe you just put the title or the author name, and you can see here the author name is coming here, and the title when it is published, and you have to save the citations, and you can do later manual changes also. You can do small things, you can do corrections. As I told, something is better than nothing, so you can use this particular tool. So you can see here from journal also I retract the references, and in text also information I am getting. And finally, from conferences, from other sources also I can retract the information. So same way how we did for web page, book and journal, same way you have to use you have to type in and you will get the information. So like silver nanoparticle, the key word I use, and I have got the information. This is the references I retrieve for the conferences. So once everything done, now what to do? What I have to do? You have to copy and paste in your Word file. Just you can save, or you can export in Word form. And in text, you can see these are the references which we have retrieved. So this is the final outcome which we want actually as a like a help to write our manuscript or thesis or assignment. This can be one of the tool which can help us. And in citation, you can just copy and paste and write. So this is the final outcome for this particular tool. And coming to the next, Microsoft Word. Every day, everyone is using Microsoft Word, but we never thought this will be the one of the tool also for references. So if you have the latest software, Microsoft Word 2010 onwards, you can get the one link that is called references. And under the references, you can see search or researcher option is there. So where you can just type in under the researcher, the word you can type in, the paper will appear, and whatever paper you want to refer, you just click onto that paper, and you click on where you want to put the citations, just click the cursor, and you will get the reference for that particular particular line, or you want to quote something, you will get. And you can edit it manually, because every software also some percentage of mistake will be there. So when you untag them, then you manually can edit it. So small things. So this is the like you can add as a source, as a citations. And so this is the final outcome which we want for our reference purpose. And there are different styles are available. You can see Harvard style is there, APA style is there. So depends what kind of the you want to refer the referencing styles. Then next is most important, which I want to highlight here, that is Zotero. And this particular is I am also using from last 5 years, and it's worth, and it's easy to get also online. Okay, it's a free of cost, no need to pay anything or anything. This is freely available. All students can use, all teachers can use. So this is a Zotero. As I told, one of the important our assistant is a Google. So we can go to Google site and we can just type in zotero.org. So this page will appear on your screen, and you can download the Zotero software. So this is the page will appear on your screen. And once you download, you will see here there are 2 things you need to download. Zotero latest updated software, and you have to download the Zotero connector. It depends on the browser, what kind of the browser you are using, whether it's a Chrome or Mozilla or what Edge. you are using that kind of the browser. Based upon that, you have to download the connector. Oh, why we have to download the connector? Then later I'll show you. So once you download the software, as usual, we have to run the software. We have to customize the software, depends on the like whether you want to go the standard or maybe customize, you can do it. And once you download, you finish it, and once you finish it in your screen, in your desktop, laptop, maybe it will appear like this. So this is the overview or the page which appear when you download Zotero in your PC or laptop. Okay, so now how to, why there is need to download the connector? So connector you can add to the particular browser. Maybe right now I am having Chrome, so I included the Zotero connector, which can connect my Chrome to the Zotero. So then once you added the Zotero connector, you can see the like this, the message will pop up on your screen, and once it appear like I want to search whether the silver nanoparticle is there. So just I type in and I can save this particular paper or the reference in Zotero software. Okay, as time is short, so I cannot go online and tell you. So this is just the information. When you go back and you can just test in, you will get information and you can do yourself also. So this is a way. And now how to include in your like a manuscript or thesis. So when you see this particular software will be integrated with your Word software, and when you open your Word, that time you will on screen, maybe I'll show you here, just a minute. In this way, like you can see here, Zotero will be integrated with your Word. So once you click in, you want to add the citation, so you have to go for the classic view. There are two things: keep source sorted and classic view. So you have to click on classic view, and once you click on classic view, whatever references you saved in your the Zotero software, it will appear here. So whichever relevant to that particular line or things, then you can just click in, and once you click in, finally it will appear, and at the end, if you keep the cursor as for your references, you just click in here, you will get the full references. Okay, so you can see here and depending upon the manuscript, the thesis style or manuscript style, I think there are about 20,000, if I'm not mistaken, 20,000 of styles are available in Zotero. Okay, so you can use it for like many. It will very helpful for publishing papers. Okay, so this is the final step. You can see you will get easily the references in your paper. You can retrieve, and moreover, you can just go through it and you will get many information. And beside this Zotero, if you don't want to download, there is another web page which can help us to find out the references, that is Zotero Vibe. It is also free, and just you have to enter the URL or maybe pub ID or something. You can just include title and you can get the references. You can see here the bibliography, and you can just copy and paste your paper. So this is another tool which can help us to speed up our work. And many more tools are there, like Autobibes are there, Dossier is there. Dossier I won't recommend you. This is just for your information, because Dossier is a somewhat like a librarian people they can use, and Dossier is a good thing. It will sort out everything, but for this actually I have not used, so I am not much sure about this, but this is also one of the important tools. I think most of the library people use this particular software. Then you can use the EasyBibe also. With EasyBibe, you can check the plagiarism also along with this. Then you can go for the bibliography. BibeMe is there. And I told Google Scholar, this is also one of the helpful tool. Like we don't want to use any software, or we are very much confused, so we can at least go through the Google Scholar. And in Google Scholar, one of the option is there. You can see the comma, and if you click on that, you will get different kinds of the styles of the paper. But here also you can copy, and some modification, minor modification if needed, you can do it manually. But it is also one of the very helpful tool which can help us to retrieve the references and write it correctly. Then PubMed, everyone knows about PubMed. PubMed also one of the option is there where you can click on site and you can get the information, and there are different formats also there. You can see here format, APA format is there. You can download in a different format also. If you don't as I told, software based, if you're not using, you can use this simple method to retrieve your references. And as I mentioned, few are they are paid version. But it's equivalent to Zotero. So I won't suggest you to go for the paid version. But if your university or your college is affording and they can give it, so it's good and well. So you see, this is another site. This is also one important good site. But here also you have to pay. But it will give a lot of information. It will retrieve from a lot of sources the information. But when it's freely available, so I would suggest you can go with the free version. So in nutshell, this is the message I want to give. Writing is either you are writing research or review article. It's a complex process where you need to accurately or concisely you need to give the information, and references why we are giving it is for which is required for reader to locate the source and information use. And citation also helps to helps the references which we are giving that is valid and concise and complete. And writing a good research, whether it's a thesis or manuscript, based upon good citations, that will that indicates the good information, and whatever referencing you are giving, it will helpful for the other researchers also in a good way. Based upon the importance of references in writing the tools, this particular topic right now in today's conference is to highlight various free referencing tools available online for writing manuscript. And whatever images and information I have used, this is just for a sharing knowledge purpose. So thank you so much all the participants for listening patientfully, and have a good day ahead. So these are the references, and I'm very much thankful to my university and the platform which I got right now to share my views. Thank you so much.
Thank you, Dr. Shivkanya Fuloria, for such a nice explanation, and the way how you have shown the examples that if somebody wants to do it practically, how it can be done in various steps. You have shown that. And especially the information about various referencing tools that can be used, for example, you have given MS Word that everybody has usually inbuilt in their desktop or in the PCs, right? And various referencing style is very, very important for our master's students, especially who are going to write the thesis. And we have noticed that sometimes the referencing may become the reason for the rejection of the manuscript if we do not cite the references properly in the manuscript. So it is one of the very, very important component of the manuscript writing, whether we are writing a research article, review article, or the manuscript, and from the student's viewpoint, if they are writing the thesis. So it is, I believe that it is something that we need to pay much more attention. And several times we have noticed that many students are not aware of this simple fact. So I think it was very enlightening for the students and the teachers as well to know about many more referencing tools that we were not probably knowing earlier. So thank you so much for your valuable time and efforts to explain the students and the faculty fraternity how to use the referencing tools. I think about elsewhere and science direct journals, they use Mendeley, right?
Yeah, one of
There's
the
a common
Mendeley
software. Yeah,
one
most of them we use that. First time you are listening to Jetiro, it's a very helpful.
Yeah, it's a very nice
Yeah,
tool. I'm
very nice. Yeah.
personally also I am using this Zotero, and last 5 years I am using, and it's freely available. Everyone can download and you utilize it.
Yeah. Very much thanks for that. Yeah, definitely we are going to use it. Till now we are using Mendeley. So thank you very much, Dr. Shivakanya. Very informative session, right? So hope all delegates, students and teachers will enjoy this session. So Dr. Sunita, question answer session, can you go? If anybody have questions, but she explained all the tools, whatever, which are available freely. So because students, they cannot purchase, even research scholars to some extent. So very informative. So from my side, so even the information what you gave is very much useful for me. Thank you.
Thank you, sir. Thank you, ma'am. Have a good day.
So even I want to thank the management, Vice Principal, St.Pauls College of Pharmacy for giving me this opportunity. It's very pleasure chairing the session of Dr. Shivakanya. Have a good day.
Thank you.
So anybody have questions, you can just post in chat box. The speaker can address you later. Thank you. Over to organizers.
Thank you, Dr. Shivakanya, for educating us on referencing and the citation tool. It was very informative.
Yes.
So my personal request is if you can send the PDF of the presentation to me. So
yeah.
When I will get time, I will go through those. Even yet, I did a lot of research in peptide field, but I am not yet habitual about automatic referencing. I do manually. I believe in manual system, but it is good to
it saves
those.
time.
Yeah, but nowadays what I'm doing, I'm using PubMed. In PubMed, those style conversion is there from there, and I'm not referencing. I'm not putting any reference which is not available on PubMed. So that's why I'm keeping the PubMed format constant. So
PubMed also one of the good tool. If you don't want to download anything, you just go to PubMed and cite. That is also one of the good. Yeah, simple.
Yes, I have noticed that when you were showing in the presentation, PubMed shows usually 4 type of referencing styles, but when we come to the Google Scholar, it shows I think 6 styles, including the Vancouver and Harvard, which we missed in the
commonly
PubMed.
used.
Yeah, so this is also one more alternative for us.
Yeah, correct. Thank you, ma'am. Thank you so much. Have a good day.
So if anybody have any question, they can put in the chat box.
Yes, sir. It's not question exactly, but I can't resist myself to express my happiness after listening to this session. So congratulations, Dr. Sukanya, madam. It's really fruitful and purposeful session. So as a convener of conference, I want to convey my thanks for being with us today, and very much enlightening and need of our session. So most of the undergraduate and postgraduate students, even research scholars also, they are not aware of all these things, most of the things what you have emphasized on. And on lighter way as an academician, so it's better to add one course, like add-on course to the students of pharmacy from the beginning itself, to customize with all these styles from the beginning. It's like whenever the whenever it is required, only they are into this field, into this awareness, like what to do and how to write and how to search and all. But we won't get much time to spend on that particular point, the supervisors and guides. So if we include this into curriculum or any add-on course kind of thing, it is really useful. Many of the even guides, they won't spend much time because they won't get much time. So, but it is really, because when correcting the things, we'll face problem. They won't follow proper style. And also we can't say that their rationale is so perfect because they won't go in deep into the literature survey and to what extent, what is the attitude towards literature survey. So these all things really superb, madam, superb session. Thank you so much.
Very nice.
Thank you, ma'am. Thank you so much.
I want to convey thanks to Raju sir also for bringing such eminent people and enlighten all our delegates. Thank you, sir.
Thank you.
So they are very enthusiastic. Sir is also there. Sir is also expert of pharmaceutical chemistry, and they are working in the same unit. He is senior associate professor there, and both are very hardworking.
Very nice.
And we visited AIMST University. There we also organized one international symposium last year when COVID was not there. So they are working very hard, and both are pharmaceutical chemistry experts. And so it is good to have friends like them, colleagues like them with us in our team.
Thank you, ma'am.
So colleagues, so here today we end our scientific lecture sessions. Remaining 3 lecture sessions will be tomorrow, but we will continue with our oral competition, which will be at 1:25 p.m. and I will take charge of that. So I ask all the 4 judges. Dr. Gaurav Gupta will not be available with us due to some emergency. So Dr. Sunita Dahiya will take charge ahead of Dr. Gupta. Dr. Ritu Kataria, Dr. Mahul Patel, Dr. Nilesh Dwivedi, all 4 judges be available at 1:25, with the similar link, and all the oral presentees. Mrs. Sudala, M. Lakshmi Prasanna, Sonu Beni, Sneha Thakur, Dinal Patel, Mohammad Sehensa, and Monu Sukla. So they should be available at that time. I will brief for 5 minutes to all the judges how to do the evaluation, and then we will continue with the oral session, and it will happen between 1:25 p.m. to 4:30 p.m. and that will end today's session, and after that we'll continue on the second day. So that is lunch time for locals. I think so. Here it is midnight, so we can take some drink. So, but thank you all for your hard efforts, and thank you all the judges for giving your valuable time and to form the scientific discussion forum. It will definitely benefit our young pharmacy professionals to solve their research project issues. So thank you very much all. So hope to see you at 1:25.
Yeah. Thank you, Dr. Razi. Thank you.
Yes, sir. Sir, along with these sessions, we have here 130 abstracts, tremendous response from most of the researchers we have received, and 12 sessions parallelly we are running, sir, here. Along with that, it's happy to share with you.
So how? So I was told that there are oral and posters in parallel. So how many orals you received?
So almost 90 above orals we have received, sir.
And
lot
posters?
Of presentations.
And posters?
And posters also around 50, sir.
49. 49.
Okay.
Yes.
Try to organize them within the time tomorrow also,
Yes, sir,
because
yes, sir. We have made
validity we have to
everything
start at
scheduled.
3
We have
p.m.
scheduled perfect.
So your
Yes, sir.
results should be ready by 3 p.m. So try to finish it today maximum, so that the result can be announced properly.
Yes, sir. Tomorrow morning also few are there, sir. But we'll make sure that by valedictory we'll be keeping every result ready.
Okay, good.
Right, sir. Thank you,
Okay,
sir.
then see you at 1:25, colleagues.
Thank you, everyone. I request all of you to have your name. Thank you.
Yes, colleagues, I welcome all of you in this oral competition. So during this session, there will be 7 orations, and those all will compete for the APP Outstanding Oration Award, which will be in the memory of Mr. Manas Tripathi, one of our colleagues who passed away. And there will be 2000 cash award for the winner of this outstanding oration, as well as the runner-up will receive 1000 cash award with medal with a appreciation award, and the remaining 5 will get the certificate of appreciation for presenting the oration. So may I ask, all our judges are available? Dr. Mahul Patel,
yes,
you are with us?
Sir, Mahul here.
Yeah.
Yes, sir, available.
Okay.
Yeah.
Okay, good. Dr. Ritu Kataria. Dr. Ritu Kataria.
Yes, sir. Yes, sir, I'm available.
Okay.
Yes, sir.
Then Dr. Nilesh Dwivedi. Dr. Nilesh. Okay. Dr. Sunita Dahiya.
Yes, I am available.
Okay. So I will call Dr. Nilesh. He will join you soon. So, okay. Over to anchor. They can introduce all the chairpersons, and only one change. Dr. Gaurav Gupta is not available. Instead of Dr. Gupta, Dr. Sunita Dahiya will take the proceeding. So over to anchor.
Good afternoon, everyone. I would like to invite Dr. Mahul and Patel. He is an assistant professor at the Department of Pharmaceutics, Faculty of Pharmacy, Dharmesh Desai University, Gujarat, India. Well, he has more than 15 years of experience in academic and research. He has completed his PhD from HNGU, Patan, Gujarat, Bachelor's of Pharmacy at BMC PER, and Master of Pharmacy from KLE College of Pharmacy, Bangalore, Karnataka. He has secured first rank in his PG examination at Rajiv Gandhi University of Health Sciences, Karnataka. He has published more than 20 research papers in international and national journals, and more than 40 presentations for his research work were represented at various international and national conferences as authors and co-authors. Well, he is approved PhD guide at Dharmesh Desai University and currently PhD scholars working under his guidance. Dr. Patel has guided 24 BPharm students, 15 M-Pharmacy students for PG dissertation research work. His thirsts are for research and micro-nano carrier-based drug delivery systems, transdermal drug delivery, and herbal formulation development. He has hands-on expertise for quality by design of experiments, statistical analysis, statistical optimization, etc. He is a life member of professional bodies such as Association of Pharmaceutical Teachers of India and Association of Pharmacy Professionals. I welcome you, sir. Next, I would like to invite.
So just an update. Dr. Mahul is associate professor, not assistant.
Yes, sorry, sir. Associate.
Okay, it's okay.
Okay.
I would like to invite Dr. Ritu Kataria. She has done her B.Pharm from Hindu College of Pharmacy, Sonipat, Haryana, M.Pharmacy from Rajiv Academy of Pharmacy, Mathura, and PhD from MD University, Haryana. Presently, she is working as Principal Director in GVM College of Pharmacy, Sonipat. She has an experience of about 13 years in teaching and research. She has published various research and review articles in peer-reviewed national and international journals. She has been awarded with Best PhD Thesis for a session 2018-19. She has delivered oral presentations in various conferences and seminars. She is a lifetime member of various associations. She has been honored as co-chairperson in various national conferences. She is author of the book entitled Pharmaceutical Marketing Management, 8th semester Thakur Publications. I welcome you, madam.
Thank you, ma'am.
Next, I would like to invite Dr. Sunita Dahiya, presently serves as Assistant Professor of Industrial Pharmacy at School of Pharmacy, University of
Puerto Rico. Puerto Rico.
USA.
It's okay.
She completed her PhD from Gautam Buddha Technical University, Uttar Pradesh, M Pharmacy from Rajiv Gandhi University of Health Sciences, Karnataka, and B Pharmacy from AR College of Pharmacy, Gujarat. Dr. Dahiya has 20 years of experience in teaching and research. She has published more than 40 articles in peer-reviewed journals and authored several book chapters with reputed publishers such as Springer, Taylor Francis, and Elsevier. She is a reviewer in international journals and editor-in-chief of Scientific New magazine, published by Association of Pharmacy Professionals. Dr. Dahiya is a recipient of grant of $30,000 from National Institute of Minority Health and Health Disparities from the National Institute of Health and $70,000 from Puerto Rico Science Trust for the research project on nanocrystals. Dr. Dahiya serves as a General Secretary of Association of Pharmacy Professionals since 2011 and is a fellow member of various national professional bodies including APP, IPA, APTI, IPGA, SOP, AAPS, and international bodies such as American Association of Pharmaceutical Sciences and American Association of Colleges of Pharmacy. Dr. Dahiya's research areas include formulation sciences, utilizing complexations, solid dispersion and nanotechnological approach for the dissolution and bioavailability enhancement of poorly water-soluble drugs and controlled release drug delivery systems. Dr. Dahiya's academic interest includes implementation of active learning methodologies such as problem-based learning, classroom seminar and journal club, and small group workshops, students' e-portfolio, and rubrics creation for assessment. Welcome, madam. Dr. Dr. Nilesh, over to Dr. Nilesh.
No,
over to Dr. Rajiv Dahiya, sir.
So you left another chairperson, Dr. Nilesh.
Sir, that Dr. Nilesh Tiwari's CV is not with us, sir. If you do
Okay, Dr. Nilesh Tiwari is the principal of Nand Kishore College of Pharmacy, Naini, Allahabad, Uttar Pradesh. And he has also served as vice principal of Sri Ram College of Pharmacy, Jabalpur, and at HOD level. So we welcome all the judges. So just some of the introduction. So you have to, one of the judge can take care of the time. So we have given 15, you have to give stick 15 minutes to the presentation and 5 minutes to the discussion. So it will be like that. So, and 5 minutes is the margin if anybody exceeds. And you have to give me an email on presidenttapp11@gmail.com after all the 7 presentations will be over, and you have to score them out of 10. I'll take the average of all and we will decide who we will announce in the valedictory who are the first, second. Okay. Is it clear to all?
Yes.
Any doubt?
No, it's clear. Yeah.
So just we can take the attendance of the presentees if they are, so that we can modify. Sudala, you are here? Sudala. Hello. Sudala Sirisa.
She's not available.
No,
Yes, sir.
Yeah. Yes.
Okay. Okay. So Dala Sirisa is available. Lakshmi Prasanna.
Yes, sir.
Okay, Lakshmi Prasanna. So you can put your video on. We want to see you. Where is Sudala Sirisa? I am unable to see.
Yes, sir.
Your video?
Yes, sir. My video is on.
Yes, you are there. Okay. Then Lakshmi Prasanna. Okay. Once you can keep, then it's okay. Sonu Beni. Sonu Beni is there? Okay. Sneha Thakur. Sneha Thakur. She's not there? Okay.
She's here, sir. No problem. I'll just.
Okay. Dr. Dinal.
Yes, sir.
Okay, you are here. Mr. Sehensa. Mr. Sehensa. Okay, Mr. Monu Sukla.
Good afternoon, sir.
Where are you? Monu Sukla.
Yes, sir.
Your video.
Yes,
okay,
sir.
You are here. Okay, good. So we are not seeing Mr. Sehensa. Otherwise, all other seems to be available. So okay. So we can start with Sudala.
Dr. Sneha Thakur also available now.
Okay.
Yeah.
Okay, no problem. So I will see if Mr. Sehensa can be available with us. So who is introducing Sudala, and we can start the presentation. Dr. Mahul, take care of the time strictly.
Sure. Okay, sir.
Okay, over to the judges.
I would like to invite Mrs. Sudala Sirisha. Her topic is formulation and evaluation of Timolol Buccal Patches. Affiliation, research scholar, University Department of Pharmaceutical Sciences, Utkal University, Bhubaneswar, Orissa. Over to Mrs. Sudala Sirisha.
Yes, sir. Thank you, sir. Yes, sir. Today, very good afternoon to one and all. So I'm very thankful to giving this opportunity to President of APP and whatever the chief persons of this oration program, Dr. Patel sir and Dr. Ritu Khatharia and Dr. Sunita Dahya. So
One second. There's a lots of echo in background. So can you solve it?
Sir, disturbances are there, sir?
Yeah.
1 minute, sir. I will solve this one. Yes, sir. Now is clear, sir?
Yeah, it is clear.
Yeah, clear.
Yeah, thank you, sir. So today, very good afternoon to one and all. Today I am going to discussing about the formulation and evaluation of Timolol Buccal Patches. And I am the research scholar from the UDPS Utkal University. And very thankful to the St.Pauls College of Pharmacy for giving this type of opportunity for the oration program. And coming to my topic here, Buccal Patches. So Buccal Patches, so coming to patches, so patches compared to the tablets and the injections, whatever the other delivery systems, this dermal delivery system is very...
Madam, you need to share your screen.
Yeah.
Okay, sir. Okay, sir. Yes, sir. Thank you.
Yes, the screen is visible, sir, everyone.
Yes, yes, visible.
Yeah, so thank you, sir. Thank you so much. So
First
coming
of
to
all,
the
it can be in a presentation mode.
sir,
Presentation mode possible hai?
हाँ sir.
No, it is not a presentation mode, ma'am.
Yeah.
It's not a presentation mode. Please do it. Please make it.
Yeah.
Yeah, now it's okay.
Now it's okay.
Thank you, sir. So coming to the buccal patches. So buccal nothing but here it is a type of a mucoadhesive system, and also the patches. So buccal patches, not only patches, we have a buccal tablet preparation we are done. So patches, coming to patches means it is a type of dermal delivery system. Compared to the tablet, that is the oral drug delivery system, and as well as the parenteral drug delivery system, these patches having the unique advantages compared to the both process. Say for example, in oral drug delivery system, when we are taking the drug in a oral delivery system, they are undergo the first pass hepatic metabolism. So that means that before reaching into the systemic circulation, they are undergo the metabolism under the hepatic system. So thereby, so many drugs having the decrease in the bioavailability. So that is the main drawback for the so many drugs when we are giving the oral delivery system. So that's why for overcoming that type of disadvantages, we are using the dermal delivery system. So many of the dermal delivery system like creams, gels, and also different types of are there. But nowadays we are going to form into the patches. So patches, not only for the transdermal delivery, we are using the buccal purpose also. Here we are using the mucoadhesive materials. Mucoadhesive materials means muco means that is a mucus, and adhesion, whatever that material we are using here, the polymer we are using here, they're having the adhesion property to the mucous membrane. So thereby there is an increase in the contacting between the delivery device with the mucous membrane. So thereby increasing the contacting. When the increasing the contacting means they're giving the increasing the systemic cell, that is a systemic delivery of the drug also. So thereby we are using this type of buccal patches, and we are using the timolol as a model drug. It is an antihypertensive drug, and it is also main drawback for that first was hepatic metabolism, and also the another drawback for here, some drugs are unstable, they're sensitive to the harsh condition of the GI tract. So in that condition also we are using this type of buccal system. So see here the introduction for the mucoadhesive system. So mucoadhesion mainly they are starting in earlier 1980s. So here I'm giving the definition: mucoadhesive drug delivery system are the delivery system which utilize the bioadhesion property of some hydrophilic polymers. See here the hydrophilic polymers, here we are using the simple HPMCE15 polymers. Hydroxypropyl methyl cellulose polymer we are using, that is the simple polymer. So that polymer having the bioadhesion property, so which become adhesive on hydration, and hence can be used for the drug targeting to particular region of the body. Otherwise that can be giving the targeted action, otherwise the local action, next the systemic action also. And the next point here, administration of the drug through the buccal route, it may overcoming the problems like a hepatic first-pass metabolism. Already I said that. And the next one is the drug degradation in the harsh condition gastrointestinal environment. So which drugs are unstable in case of acidic condition of the GI system. In that cases, we are giving this type of system. And also it is main advantages to the patient is the self-administration process, self-medication. If you want the drug action, you can place the medicine at your cheek. Otherwise, we are not need any medication this time. Just we have to remove the tablet or remove the patch from the site of action. So this is a very simple self-medication process. And this is a next one is the structure of buccal mucosa. So it is a human mucosa. They're containing the superficial layer, that is the mucus. So that mucus can be secreted by the submucosal cells which are present at the bottom. So this mucus, it is a viscous nature. It is a very viscous nature, and they are containing the main glycoprotein, that is the mucin. So when the viscous nature is mainly responsible by the mucin molecule. And this main barrier for the drug diffusion also. Why? Because it is a viscous nature. So that's why that can hinders the drug passage also. And next, the second figure I will show here how the drug can be transferred from the mucous layer to the systemic circulation through the transcellular process and the paracellular process. Paracellular nothing but that is the between the gaps of the cells, and then transcellular nothing but that is across the cell. And next one, here the mucus is it is the first barrier, and also they are containing nutrients and so many compounds can be compulsory passing through the mucous membrane. So this is the main barrier for the buccal patches. And next one, coming to my topic, that is I'm taking the timolol. So timolol, it is a beta adrenergic antagonist, and they having the shorter half-life, that is a 2.5 to 5 hours. And also they are having the bioavailability is a 60%. So here the one point here, when it is having the shorter half-life, that means compulsory we are giving the repeating dose. We are giving the repeated dose. So for that purpose, we are using the extended release tablet. But in case of extended release tablet also, they are having the only bioavailability is a 60%. So for increasing that bioavailability, we are using the buccal patches. We are using the buccal patch. So here the present work mainly formulating the mucoadhesive buccal patch for the timolol using the different concentration of HPMC E15 polymer, that is a hydrophilic polymer, to circumvent hepatic first pass metabolism, and also to prevent the degradation in the gastrointestinal part. So every formulation, whatever that formulation we are going to start, compulsory we have to check whether there any interaction between the drug and as well as the polymer. That is important in any case. Why? Because if any interaction is there, either it is a physical degradation, either chemical degradation can be occur. So compulsory we have to start the experiment with the drug compatibility study. So here we are conducting the FTR study. So for the physical compatibility purpose, not chemical compatibility, it is a physical interaction purpose. So here just we are taking the pure drug and also the pure HPMC polymer, and we are conducting the FTR study, and those peaks we are comparing with the polymer and drug mixture. So based on that peak, we are concluding that there is no interaction between the drug and polymer. And next, after that, when it is there is no interaction, then we go for the patch preparation. Okay, it is a next step. But when you are go for the patch preparation, compulsory you have to do the one step, that is whether the drug you are selecting that can be passing through the mucous membrane, or we have to check. If it is not possible, we are not going for the patch preparation. It is unnecessary. When it is passing through the buccal mucosa, then it is easily we have to go for the patch preparation. So before going to this patch preparation, we are conducting the permeation study by using only simple drug solution. Drug solution. So based on that study, it is concluded that the drug easily passing through the buccal mucosa. And after that, we go for the buccal patch preparation. It is a very simple preparation by using the solvent casting method. So here just we are taking the 2 test tube. In a 1 test tube, you are taking the solvent mixture. Here we are using the solvent is a dichloromethane and as well as the methanol in a 1:1 ratio. In that we are dissolving the polymer to form a viscous solution. And in another beaker, another test tube, we are taking the drug and plasticizer in a solvent mixture, and both are you have to dissolve, and you have to wait for the 6 hours for the swelling of the polymer. After swelling is over, then the drug solution can be added to the polymer solution and again shake it, and then you can stay for the sonication process. Why we are using the sonification process here? During the shaking or during the mixing, any air can be entrapped in a polymer mixture. So that entrapped air will be removed by application of the sonication. So after sonication, the air bubble can be removed. Then that can be poured into the Petri plate, and you can dry it for the overnight period, and then you are getting the buccal patches like this. We are getting the like a neat and without any imperfection, without any cracks, and without any precipitation, we are getting this type of patch. That is a transparent patch we are taking. And after that, see, buccal patches preparation, it is 3 types are there. That means based on the diffusion process, based on the drug release direction, it is a bidirectional way, multidirectional way, unidirectional way. See, the first one is a multidirectional way. That means we are not using any baking membrane for the restricting the drug release from the device. So we are not using any baking layer in the type 1. In a type 2, we are using the baking layer for the minimizing the bidirectional release of the drug. And the third one, it is a different way. That means all the sides, only one side we are not covering. On all the sides we are covering with the baking membrane for the unidirectional purpose. So which one we are using? So based on that, we are preparing the buccal patch. And also the tablets is also similar, but here in the tablets we are using the compression method, but here in the patches we are using the solvent casting method. And next one, it is a formulation table. I am taking the ratios of different concentration of the reagent and polymer, and based on that, this is a DCM and methanol, that is a solvent mixture. We are taking the 1:1, and the propylene glycol here. The propylene glycol, it is the most important here. It is a plasticizer. It is a plasticity. So for giving the patch, for giving the plasticity to the patch purpose, we are giving the propylene glycol. And after getting this patch, each patch having the size is a 3.14 square centimeter, and the 3.14 square centimeter of patch contains the 10 mg of timolol. So based on that, we are calculating, and based on that, we are preparing the patch. And after that, we are getting the neat and clean patches. Then we go for the evaluation parameter. So the patch compulsory, we had to use the checking the, it is a smooth texture, otherwise it is the elegant, it is the uniform weight and thickness, and also the uniform drug content. So these are the normal physical chemical properties we are using. And another one we are using, that is a folding endurance. It is a basic property for the mechanical strength, to check the mechanical strength of the patch. And next point is here, pH. Why we are using the pH? Why we are determining the pH here? We are using the buccal patches. So that means you can insert the patch in a buccal portion. So the buccal pH is equivalent to the our patch preparation pH also. Patch pH is similar to the buccal pH. It is necessary. If the pH can be altered, what happens? That can giving the irritation to the our buccal mucosa. So that's why we had to maintain, we had to prepare the patches based on the pH, that is you should maintain the 6.5. And also we are conducting the surface pH studies by using the agar medium.
Sudala, I think 2 minutes remaining. So try to conclude your presentations. Yeah.
Okay, sir. Okay, sir. Thank you. So this is a moisture absorption. Why? Because we are using the hydrophilic polymer. So that's why we should conducting the moisture absorption. And the mechanical properties, that is the tensile strength and elongation break, by using the tensile strength apparatus we have in our university. By using that, we are conducting this one. And another one is and next one is a in vitro release studies. So by using the USB type 2 apparatus, just we had to modifying the application of the device in the basket. So for that purpose, we are using the glass slide. In that glass slide, we had to fix that patch, and that can be inserted into the dissolution apparatus. And we are conducting only 6 hours, not 24 hours, only 6 hours. And next one is this is important ex vivo permeation studies. We are using the pig mucosa. So the pig mucosa, it is having the similar property to the our human buccal mucosa. So that's why we are using the pig buccal mucosa. So before going to study, compulsory we are going to slaughterhouse, and then we are taking the buccal portion of the pig, and then we can separate the mucosa like this. And then that can be mounted in a French diffusion cell, and then conducting the ex vivo studies. And then conducting the ex vivo studies. And coming to conclusion here, so compared to the tablets, my intention, my view is, compared to the all other delivery system, It is a simple process. Patches is a simple process. We are using the simple polymers with a cost effective, and it is also patient convenient compared to the parental system also. It is the painful to the patient. Every time we are giving the injection to the patient. For example, in case of cancer patients also, they are feeling the very pain to the body when they are giving the chemotherapeutic drugs. So in that cases, you are giving the pain relieving patches, like a patch you can giving at the skin. It is a very simple, and it is not harmful to the patient also. When they are not required, they are easily removing the patch. So my intention, it is a very cost effective, and it is a patient convenient, and also it is a very simple process to prepare the patches in a laboratory. So this is my study, sir, and then we are recommending and we are continuing this study to up to in vivo studies, and we are using the guinea pigs. So to the guinea pigs, we are administered these buccal patches, whatever we are preparing, and then we are conducting the in vivo studies also. These are the references. And thank you, sir. Thank you for this giving the wonderful opportunity to my PhD career. Thank you so much, sir.
Thank you, Ms. Sadala.
Yeah.
Yeah, so thank you, sir.
Okay. I have a question. You have selected HPMC E15 as the mucoadhesive polymer, right?
Yes, ma'am.
How do you see the mucoadhesive properties of this polymer with context to the retention time of your pages?
Yeah, ma'am. Actually, we are conducting. So before selecting the polymer, also we are doing the mucoadhesion strength. Also, we are detecting. So by using the buccal mucosa, also, just we are taking the. Yes, madam. Actually, we have a one calculation also, madam, in vitro bioadhesive studies. So in that cases, just normally we are preparing the patch, and also we are taking the buccal mucosa. They are having the ultra testing in our laboratory. We have a ultra test make machine best success UK equipment is there, sir. So in that equipment, two cylinders are there. In a one cylinder, we are placing the patch, and another cylinder we are placing the mucous membrane. So just by using the some force every second for the movement 0.5 newtons we are applying. So at what force the mucus can be attached to the polymer, and at what force the mucus can be detaching from the polymer. So that can be we are detecting. So based on that, we are selecting some polymers here.
That means you have also tried with another polymers like Citation?
Yeah, ma'am. Yes, Citation and also the sodium alginate, and also different grades of HPNC also, ma'am. HPNC E5, E50 also we are trying. But
okay. One more point that I want to add. Why you have selected low viscosity HPMC?
Yes, sir. When we are using the high viscous, what happens when the thickness is also increases? When the high viscous polymers we are using, that means the molecular weight is also increases. When the molecular weight is increases, the thickness of the patch is increases. When the thickness is increases, one is diffusion can be affected, and as well as the patient is also feeling the discomfort. That means they are having the like a one point in my buccal cheeks. There is a one thing is there, They are having a like a feeling. So compulsory we have to reduce the thickness purpose. We are using the low viscous and the low molecular weight of polymers.
Okay.
Otherwise, when we are using the low viscous and also the low amount of polymer we are using. So when we are using the low amount of polymer, what happens? The drug will be precipitated. The sufficient polymer is not observed, not is present. So that's why what happened? The drug will be precipitated. And that can be affecting the stability of the patch.
Okay. And your system is bidirectional or it is a unidirectional?
Yes, sir. Unidirectional, sir. We are coating with the ethyl cellulose, sir.
Okay, yeah.
Okay, I have one more question that you have shown the in vitro release studies, right?
Yes.
So you have used USP apparatus too, right?
Yes, madam. It is a modified, madam. Actually, we are using the USB type 2 apparatus, but in that we are giving this some modification. That means just simple, we are using the glass slide. In a microbiology lab, we are using the glass slide. So that glass slide we are using, and that just we are attaching the cyanoacrylate plaster is there. That is a two-side plaster is there. That one side we are stick to the slide, and another side we are placing the patch, and that totally we are covered with the gelatin membrane, and that we are placing in a basket.
Have you also shown the release mechanisms in terms of the mathematical modeling, like other?
Yeah, ma'am.
So,
We are connecting every PEPAS model and zero order. It is a zero order or a first order. It is a PEPAS model or a Gucci model. Everything we are done, ma'am. Actually, it is following the zero order, ma'am, and also it is following the diffusion process, ma'am.
okay, Hikuji matrix.
Yeah, ma'am.
Okay, thank you. The only one suggestion I have is you should have taken a little less time for the initial introduction about the oral drug delivery and that, so that we can get more. And this is my recommendation for all the participants of the oral presentations, that if you please give little less time for the introduction, we can have more time on the research work that you want to do and more fruitful discussions. If Dr. Mahul and other chairpersons have any questions.
It's real. That's why I have interrupt during the presentation, that we should focus more on that part, research part. Yeah.
Sudala, I have one question, that any such kind of buccal patches are available in market?
Yes, madam. Marketed preparations. So many marketed preparations are there now. Available is there.
Can you name?
Yes, madam. I have 1 minute, madam. Actually, I have a list.
I think you should compare your buccal patches with those marketed preparations, and that's why I am asking about the same.
But here in a Timolal, I'm not using any marketed preparation, madam. Actually, it is a one side part of my research work. Actually, my work is totally relating to the transdermal drug delivery system, madam. I'm interested in ionotophoresis.
Okay.
It is
okay. All the best. All the best.
Thank
all
you,
the best, Shraddha.
Thank you.
Next participant.
Next participant is Mrs. Sudala Sirisha, ma'am.
No. Next one, Miss Prasanna.
Miss Lakshmi Prasanna. Her topic is formulation, development, and evaluation of fluvastatin in situ gels. Affiliation, research scholar, Department of Pharmaceutical Technology, Visakhapatnam, Andhra Pradesh. Andhra University. Over to you, ma'am. Miss M. Lakshmi Prasanna.
Please allow my screen share. Thank you. A very good afternoon to one and all. First of all, thank you everyone give me this opportunity. Myself, Prasanna, doing PhD part-time in Andhra University, working as Associate Professor in VJS College of Pharmacy, Rajahmundry. I'm going to explain about my research on formulation, evaluation of fluvastatin floating oral in situ gel. And these are the contents I'm going to explain here. And main aim of this study involves formulation and evaluation of floating oral in situ gel containing fluvastatin, and this study is formulate oral containing fluvastatin polymers and perform pre-formulation studies of pure drugs and conducting excipient compatibility studies, then draw standard calibration curve, and then prepare the oral in situ gelling system and then evaluation. So these are the plan of work in this my study. And gel solution before administration in the body, but once administered inside the body, and goes dilation in situ for a gel. So what is the advantage of this in situ gel administration? Increase focal bioavailability, reduce dose frequency, improved patient compliance. Its production is less complex and lowest the investment. And administration routes for in situ gel, take an oral route and also use it to ocular, rectal, vaginal, injectable, and intraperitoneal routes can be used these kind of in situ gels. And what is the approaches to produce in situ gel is different approaches and mechanisms utilized involved in this producing in situ gel formation based on producing physical changes, based on producing chemical changes, based on physiological stimuli, dilution sensitive, electrical signal sensitive, light sensitive, approaches to produce gel. Mechanism in situ gel is liquid solutions for administration, but gel under physiological conditions. So physiological conditions, by using some dermally triggered system and pH sensitive polymers, we have to go into use. So ionic cross linkage, pH change, temperature modulation, polymer solutions of pectin, sodium. And complex with the spin citrate, so they are breaking the environment of the stomach to release free divalent ions. With the water. And coming to applications, oral drug delivery, delivery, ocular drug delivery, dermal delivery, nasal drug delivery, application. So regarding drug profile, so this is the
Madam, your voice is not audible, madam. Sorry for interruption. Your voice is not audible. And even your that video is turned off. Please turn on your video. As I'm not a chairperson of this session, but it is my responsibility to say it.
Actually, this one automatically. So now it is visible method. Voice is good method. Can you hear my voice?
Still not audible. Yes, you can continue. No problem.
The present, sir?
Yeah, you can continue your presentation.
Okay, sir. So this is the drug profile of my study. So molecular structure and chemical formula and molecular weight. And category of the drug is anti-hyperlipidemia. And these are the excipients I'm using in this research work: sodium alginate, calcium carbonate, sodium citrate, HPMC, K4M, and Gargum, HPMC, K15M. And these are the list of materials and equipment I'm using. So coming to experimental work, I'm starting with pre-formulation studies, solubility studies. So solubility of fluvastatin was carried out in different solvents. Initially, I'm tested on methanol, ethanol, 7.4 pH, and 6.8 pH buffer. So among which all the good solubility will be shown in the 0.1 normal HCl, and then after determination of absorption maximum by UV spectrophotometer. So these are the plots I'm giving here, and preparation of calibration by so different stock solution I'm preparing by using the different concentrations 0.1 normal HCl. So these are the absorbance of absorbance I'm noted at 267 nanometers. So and then after I'm conducting drug excipient compatibility studies between the drug and excipient. So there is no incompatibility between that. So what I'm using, those will be compatible. So this is the formulation table, and totally I'm performing 12 formulations. So formulation 12, I'm using drug and along with these kind of excipients. And coming to method of preparation, in situ gels, fluvastatin is a drug, so that I'm preparing composition of the drug according to the table. So 100 ml of the beaker I'm using. So in that sodium arsenate and added with other polymers mixed with the 60 ml of distilled water and heat the mixture up to 60 degrees centigrade solution. By using the heat magnetic cylinder, so take the another beaker 100 ml and to that add sodium citrate and calcium carbonate, then mix with water and heat the mixture at 60 degrees till the solution occurs. Now take another beaker, add 5 ml methanol and with the drug. So then 3 mixtures are mixed at 60 degrees. After cooling the solution below the 40 degrees centigrade, heat the above mixture in mechanical stirring for 30 minutes. So well to get the final preparation which was stored at the umbilical bottle. So finally, I'm going to evaluate various parameters, drug content. So these are the studies I'm showing here, and in vitro chelation study also I'm performing. So gelling studies were carried out using 0.1 normal HCl. So and after that viscosity study, these are the results. And in vitro floating study, so in vitro floating study was carried out by introducing 5 ml of formulation into a beaker containing 100 ml of 0.1 normal HCl. pH 1.2 at 37 degrees without much disturbance. So the time of formulation constantly floated on surface of the dissolution media. So that is one recorded. So from F1 formulation to F12 formulation, I'm doing this same manner. So this is the floating time I'm giving here. And finally, I'm conducting in vitro drug release studies from F1 to F12. So F8 will be showing the more sustained action, and these are the results, and follows the zero order and Higuchi, and finally for the optimized formulation conducting stability studies, and they will be stable over the 3 months period of time. And finally, I'm concluding my topic. Fluvastatin oral in situ gelling system were prepared by using these all the polymers Gotham, HPLC, sodium HPNC K4M, HPNC K15M, sodium citrate, calcium carbonate, and sodium alginate. So total of the 12 formulations were prepared, and F8 was found to be the best formulation with the Gargam polymer, and the drug and polymers were subjected for compatibility study using FPR studies, so which revealed that there was no interaction between the drug and polymer. So the prepared formulations were evaluated for drug content, floating lag time, total floating time, viscosity, gelling nature, visual appearance. In vitro release studies were also performed. The in vitro release studies of all the formulations, among them F8 formulation containing Gargam shows drug release of 98.08% by the end of the 12-hour study. So the release scientific of the optimized formulation was best fitted into the Higuchi model and showed zero order drug release. So from the above experimental results, it can be concluded that fluvastatin was chosen as a model candidate for development of oral in situ gel. So since they possess near ideal that these drugs must have formulating sustained drug delivery. Thank you, sir.
Thank you, Ms. Prasanna.
Thank you, sir.
Yes, Sunita ma'am, any questions?
Yes, you did the in vitro dissolution of the gel, right? How did you do
Yes.
it?
By using USP2 dissolution apparatus, ma'am. So here I'm using 0.1 normal HCl is a buffer. So in that prepared sample I'm adding. So that we have to performing the 12 hours dissolution studies. So take the sample for every 1 hour period of time and check the absorbance value and calculate the percentage release of the drug.
Okay.
I'm asking in terms of the formulation because it is in situ gel, right?
Yes, ma'am.
So
How you
when
have
you
added that? Yeah, how you have incorporated that particular formulation?
Yeah, this is a temperature sensitive polymer, temperature sensitive. So that I'm creating temperature similar to the body temperature conditions, whatever the dissolution medium I'm using. So that in that medium that will be set as a gel.
But whether the mechanism of gelation is pH responsive or temperature responsive?
pH, the temperature responsive, ma'am.
Not pH?
No.
Not pH. By administration of the drug inside the body due to temperature condition that would be set as a gel mask.
And it's intended for the oral administration?
Oral, ma'am. Oral, madam. Floating oral solution.
Okay.
In situ gel.
Okay,
Floating
but I think when you have selected the guar gum, then I think it may not be as I think temperature sensitive. It may be temperature sensitive, but I think pH plays a very important role, or maybe the ionic
Okay.
sensation.
Okay, sir.
That is what my opinion.
Okay.
That it maybe work well with the, especially you have mentioned the mechanism also, that
Yes, sir.
in presence of ion it's converted into the gelling system.
Yes, sir.
Yeah, it's a good suggestion because you can also gather some data.
Yes, sir.
You have used various polymers. So
Okay, you have good
yes,
compositions.
Sir, I will
Yeah.
study in this. Thank you, sir, for your suggestion. And if I'm doing in that way also, so that if it is showing, so both I'm giving the results, sir. It will be helpful.
okay, thank you and best wishes.
Thank you. Thank you, ma'am.
आपको शेयरिंग आता है ना? उसको पहले खोल के रखो.
Next, I would like to invite Ms. Sonu Benny. Her topic is Designing of MCT4 Inhibitors, a novel anti-cancer weapon to arrest the cancer cells and their microenvironment. Affiliation, Research Scholar, Department of Pharmaceutical Chemistry and Analysis, Amrita School of Pharmacy, Amrita Vishwa Vidyapeetham, AIMS Health Sciences Campus, Kochi, Kerala. Over to you, Ms. Soni, madam.
Thank you so much. Can you share the screen?
Yes, madam. We share the screen, madam.
Is it visible right now?
Yeah.
Yes, madam.
Am I audible? Can I start, sir?
Yeah, you can start.
Yes, you can start.
Okay. A warm good afternoon to one and all. Myself, Sonu Benny, PhD Research Scholar, Department of Pharmaceutical Chemistry and Analysis, Amrita School of Pharmacy, Amrita Vishwa Vidyapeetham, Cochin, Kerala. I'm so happy to present my research work entitled Designing of MCT4 Inhibitors, a novel anti-cancer weapon to arrest the cancer cells and their microenvironment. To be brief about the background of the study, according to Dr. Otto Heinrich Warburg, the Nobel laureate, he has found that no disease can exist in an alkaline environment. So according to him, acidic environment is the root cause of cancer, and he found this as the root cause of cancer, and he was honored with the Nobel Prize for this discovery of the root cause of cancer. During his investigation, he found that there is an unusual metabolic behavior has been exhibited by the cancer cell in comparison to the normal cells. That is, when the normal cells are following mitochondrial oxidative phosphorylation in the presence of oxygen, and in the absence of oxygen, they will be following anaerobic glycolysis. But in the case of cancer cells, irrespective of the presence or absence of oxygen, they will be following aerobic glycolysis, and this phenomenon has been termed as Warburg effect. So this is the invention in the year of 1920. And later, over a century, so many discussions has been conducted with the same topic, and in the year of 2009, a modified concept of the Warburg hypothesis has been put forward by Dr. Stefanos Paldives and a group of scientists. According to them, this reverse Warburg effect, this is a two-compartment metabolism. In the case of reverse Warburg effect, that is the metabolic switching. But in the case of reverse Warburg effect, they will propose that this is a two-compartment metabolism, which is explaining a metabolic symbiosis or a metabolic coupling existing between the cancer cells and the cancer-associated fibroblast. So, as per them, it accounts for the existence of tumor microenvironment, and also it states that there is the possibility for having intratumoral heterogeneity in the cancer cells. So they are stating the occurrence of this tumor microenvironment as a reason for the cancer progression, recurrence of the cancer, and the poor clinical outcomes. Here, we can see that lactic acid, as a result of this cancer cell metabolism, lactic acid will be generating from the cancer-associated fibroblast, and this lactic acid will be deposited in the extracellular space. This lactic acid will be taken up by the cancer cell as one of the prominent metabolic fuel. So here what is happening is that apart from being an important metabolic fuel, this lactate is having so many roles in the tumorigenesis, including they are causing immunosuppressions by inhibiting the activation of the T cells, and also metastatic dissemination, therapy resistance, resistance to the programmed cell death, including apoptosis anoicus. Then also it is responsible for creating an acidic tumor microenvironment, which is helping the condition of cancer and its progression. So what we are adopting here, the research strategy is that here we can see that lactic acid will be generated from the cancer-associated fibroblast, and it will be released to the extracellular space with the help of the receptor MCT4, and from there it will be taken up by the cancer cell with the help of the receptor MCT1. So here we are going to focus the receptor MCT4, that is the lactate exporter. So MCT4 will be exporting lactate from the hypoxic cancer cell and the cancer-associated fibroblast. So once we are inhibiting that particular receptor, what we can do is that we can prevent the export of lactic acid from the cancer-associated fibroblast to the extracellular space. So there will not be any lactic acid that have to be taken up by the cancer cells. And also this lactic acid will be causing a condition of lactic acidosis, and with that we can destroy that tumor microenvironment, which composed of the cancer associated fibroblast, hypoxic cancer cells, and the lactic acid that is generated and deposited in the extracellular space. So when we are focusing on the monocarboxylate transporters, the literature reviews are giving us the support that it is having expression in most of the human organs. So the aim of the research is to design a new small molecule to target both the cancer cells and its microenvironment through the inhibition of MCT4 receptor. And the objectives are the artificial neural networking and the machine learning to identify and evaluate this novel target, then the homology modeling and the evaluation to construct a valid 3D protein structure of MCT4, drug designing of new small molecules to inhibit MCT4 receptor. So this is the research work in total. So moving to the methodology, the major methodologies that we adopted here are the neural networking analysis, evaluation of the target, homology modeling, validation of the protein structure, and the drug designing. Moving to the results and discussion. Because by the help of the cytoscape, we generated a neural networking, artificial biological neural networking for the particular protein MCT4. And we found that this is one of the protein that is having interaction with many of the proteins in the neighborhood. So this SLC16A3, this is the target of interest, that is our MCT4. And here we can found that string analysis, that is the analysis which help us to identify the protein-protein interactions. So this is the protein networking of MCT4, and the major observations are it found to be having co-expression with basagin, that is the ancillary protein helping in the transport of lactate, LDHA, that is one of the protein that helps in the conversion of lactic acid. Many of the other protein members of MCT, monocarboxylate transporter family, they are SLC2A1, SLC17A5 and all. Then also it is having activation along with VEGF, that is the vascular endothelial growth factor. So then also it is having networking or connection with carbonic anhydrase, that is helping to have an acidic tumor microenvironment. So altogether, the string analysis is giving us an impression that it is having close contact with many of the proteins that is having a significant role in the cancer metabolism or in the various phases of the tumorigenesis, including proliferation and other mechanism, other phases of the cancer. This is the stitch analysis report. Stitch analysis is giving an idea about the protein chemical interactions. And here we can see that our desired protein is having interaction with lactate, diacetate, pyruvic acid. So many chemicals or the various metabolites present or including in the cancer metabolism, they are having interaction with our particular protein. So it is also giving us the same impression that it will be having a role in the cancer metabolism. This is the mRNA expression, gene expression, and protein expression predictions done by the Human Protein Atlas and the GTEx tool. So it shows us that the protein MCT4 is having medium to high expression in almost all the human organs. This is the target disease network generated by the open target software. So here we can see that there are large bubbles. Glioma, neoplasm, cancer. So all these will be the area where MCT4 can provide a significant therapeutic action. This is the disease tree network generated by the open target software. Here we can differentiate the intensity or the possibility where it have produced the strong therapeutic action. It can be distinguished on the basis of the intensity of the color. So here we can see that it will be having a prominent action in various cell proliferation disorders, including neoplasm, cancer, glioma, and the glioblastoma. This is the metabolic pathway prediction done by the KIC pathway database. And here we can see that the one-line prediction is that it will be having a major role in the central carbon metabolism in the cancer cell. This tool is helping us to have an idea about the possible pathway in the normal cell as well as in the cancer cell. So in the case of normal cell, it shows that it will be transporting lactate that is generating as a result of mitochondrial oxidative phosphorylation. And in the case of cancer cell, we can see that it will be having close association with various pathways or various metabolisms involved in the cancer cells. This is the possible pathways predicted by the pathway commons tool. In specific, they are saying that it will be having based in interaction, then proton coupled MCT or the monocarboxylate transport, and also it will be having an effect on the pyruvate metabolism. So altogether, on the basis of this artificial intelligence and machine learning evaluation, we are having an impression that it is MCT4 is an interactive protein that is having association or connection with many prominent proteins and chemicals involved in tumorigenesis and various cancer metabolism. Also, it is having good expression in the human organs, and the possible pathways are also supporting our findings. So MCT4 can be a potential candidate for the novel target-based anti-cancer therapies. So with this impression, we move to the homology modeling to construct a valid 3D protein structure of the MCT4. So it starts with the collection of amino acid sequence from the UniProt database. Then we had performed the BLAST-P search for finding out the most similar amino acid structure to construct a valid 3D protein structure. Then we had performed the homology modeling by the template sequence, target template sequence alignment will be done there. Then we will be looking for the stability and other properties of this protein molecule. So here we can see that 3 of the amino acids are present in the disallowed region or the unfavored region. So it shows the stability issues with this particular protein. And on the basis of this Ramachandran plot analysis, we will be performing loop refinement and energy minimization. Then after that, in the right side, we can see the second diagram that has been obtained after performing this energy minimization and loop refinement. So here we can see that all the amino acids are present in the allowed region only. So it is a stable protein structure. Then we had performed the mole probability analysis to quantify the amino acid concentration in the allowed region, highly favored region, and the disallowed region. This is the PROSA analysis, protein structure analysis, that has been conducted to find out the quality of the protein model, then the energy distribution of the protein model, and the quality score of the protein model. So we got a protein structure. Here in this study, we had selected 3 top amino acids that is having close amino acid sequence to that of our MCT4 or the target amino acid. Then here we can see that this is the results of the PROSA analysis, the quality and the energy distribution. In the case of stability, we can see that blue color regions are representing the lowest energy, so that it will be the highest stable one, and the red part is highest energy and the low stable one. This is the mole probability software results that shows the quantification of the amino acid concentrations in various regions as per the Ramachandran plot. This is the protein reliability report. So here in the left side, the report that is generated immediately after the homology modeling. So here we can see that so many problems are there in association to our protein structure, including unusual beta factors and all. So we had performed the loop refinement, side chain refinement, and also energy minimization. And after that, we got a reliability report for our protein, and that is displayed in the right side of the diagram. So here we can see that almost all the problems were solved to a limit, and now we are having a comparatively reliable protein structure. This is the binding site prediction done by the sitemap module. Here we can see that homology modeling has been done. So we got a 3D protein model. So here we have to find out which will be the possible binding cavities or pocket where we have to bind our ligand. And for that binding site prediction has been done by using sitemap module, and we find out the possible pockets. In order to identify and to validate this newly constructed 3D protein structure, we had performed the molecular docking studies also. So altogether, we got a stable, reliable, and sufficient quality, energy distribution, molecular docking studies, and interaction. Altogether, we selected the better PDB structure of MCT4 for the further drug designing studies. In the drug designing part, first of all, we generated a ligand library by selecting the non-specific MCT inhibitors. At present, no inhibitors were available for the MCT4 receptor, so we had selected other MCT inhibitors. And also then we performed, this is the fragment-based drug designing approach. Then we generated fragmentation. So all the possible fragments were generated. It were around 6,000 fragments were there. Then the development and screening of new hits has been performed by clustering, growing, and also we checked for the binding affinity of each of the hits. Then the evaluation has been done to find out the top hits. These are some of the MCT inhibitors, non-specific MCT inhibitors from our library. And these are the molecules. So we perform the fragmentation from 6,000 fragments. We perform high throughput virtual screening, and we got the top hits. The second approach was the machine learning or the ILP-based QSCR approach, that is the machine learning assisted pharmacophore-based drug designing approach. Here, by giving the inputs, we got a prediction has been done by the software for the possible pharmacophore, and this pharmacophore will be used to screen the possible ligands from the various ligand libraries, and it will be like a drug repurposing study on the basis of this pharmacophore-based drug designing. So altogether, after the study, we got some coumarin derivatives, chalcone derivatives, and amino carboxylic acid derivatives from the fragment-based drug designing. And from the QSCR-based pharmacophore-based drug designing, we got coumarin-containing molecules, a pharmacophore we got from that one. So we will be using the same for the drug repurposing study. We will be screening the same against the drug library, and we will be finding out the suitable ligands. So altogether, the better MCT4 ligand will be selected, and that will be further proceeded with the synthesis. So altogether, as a conclusion of this drug designing study, MCT4 gene was found to be an interactive protein, which is having interaction with the nearby proteins that is significant in the cancer metabolism. Homology modeling has been performed, and we got a valid protein structure, 3D structure of MCT4, and we evaluated the same. Then fragment-based and pharmacophore-based drug designing has been done, and novel inhibitors of MCT4 has been derived. In the future perspective of this study, further screenings, molecular dynamics, QM/MM studies, free energy binding studies will be conducted, and also synthetic feasibility will be checked for these compounds, and we will be going with the chemical synthesis of the same MCT4 inhibitor. These are a few of the references. That's all from my part. Thank you, ma'am. Thank you.
Sonu, it was a wonderful presentation. Just I want to ask some questions. That which software you use for the molecular modeling?
For the, ma'am, is it homology modeling, ma'am?
Yes, ma'am.
For the homology modeling, we use Schrodinger, prime module of the Schrodinger.
Okay, Schrodinger. And
yes, ma'am. And for
for
the
site mapping?
Site mapping
For
is
site
also, yes, ma'am.
that is available in the same software,
yes.
right?
Yes, ma'am.
Okay.
And
Okay.
PROSA, mole probability, they are the other software. We have used the same for the evaluation.
Okay. All these docking you have done yourself?
Yes, ma'am. This is my ongoing PhD work, ma'am.
Okay, okay, okay. So how you explain this homology modeling?
Sorry, ma'am.
Are you sure that the protein you have selected is, are you sure about this protein?
Yes, ma'am. Actually, a preliminary study has been conducted with the Swiss PDB modeler, Swiss modeling software. So there also we got the same result. Then we proceeded with the Schrodinger. Yeah.
Okay. You have not selected your final lead.
Yes, ma'am. No, ma'am. Some studies are still going on with that. We are having some issues with the software in between.
Okay, okay. Along with these parameters, are you looking for the ADME properties and the quick prop simulation studies, toxicity studies, these all parameters, and the Lipinski rule of 5, these all parameters you have to look for the selection of the final lead, right?
Yes, ma'am.
Okay. Otherwise, it was a nice presentation. Lots of data you have collected till date. Good. All the best.
Thank you so much, ma'am.
Miss Menu, Miss Sonu Benny, sorry.
Yes, ma'am.
It was an excellent presentation, and although you have answered almost all questions, and Dr. Ritu Kataria has asked some more, but I have only one information if you can provide. You mentioned that the MCT4 receptor is overexpressed, rather expressed in most of the cancers, right?
Yes, ma'am.
Do you have any information that in which particular cancer it can be, we can predict that it can be the most useful based on its overexpression?
Yes, ma'am. Actually, this MCT4 has been used as a biomarker in various types of cancer. So when we are going to the literature studies, mostly the same has been used in colon cancer and in glioblastoma. These two are and also in the breast cancer. So these are the mainly seen one according to the literature reviews, ma'am.
Okay. Thank you and best wishes.
Thank you so much, ma'am.
Madam, please stop your screen sharing.
Okay.
Any questions? No more questions. Okay.
Now I would like to invite Dr. Sneha Thakur for presenting on topic facile synthesis of bovelic acid silver nanoparticles in silico docking, ADMET studies on SARS COVID proteins. Affiliation to Associate Professor, Department of Pharmacognosy, St.Pauls College of Pharmacy, Turkamjal, Rangareddy, Hyderabad. Over to you, madam.
Madam, please stop your screen sharing.
Yes, sir.
Yeah, thank you.
A very good afternoon to the dignitaries and all the pharma fraternity. This is Dr. Sneha Thakur, Associate Professor, HOD, Department of Pharmacognosy, present here to present my research work entitled on facile synthesis of boswellic acid silver nanoparticles along with in silico docking ADMET studies on SARS-CoV proteins. These are the contents of the presentation.
Madam, your
As
slide
we
is
all know, nanotechnology is an emerging trend, and green synthesis is the best method to synthesize herbal-derived nanoparticles, which have.
Yeah, Dr. Thakur, your screen is not moving.
Can you make it to presentation mode, please?
Yes. Just so now, sir, can you see the screen?
It is not yet in presentation mode.
It is not in presentation mode.
Just slideshow or whatever you want.
I'm sharing the slides, sir. Now?
Yeah, yes, okay. Okay.
Okay.
Now it's visible. You can continue.
Yes. So these are the contents of the presentation. As we all know, nanotechnology is an emerging trend, and green synthesis is the best method to synthesize the herbal-derived nanoparticles, which are having a potential future in eliminating the problems associated with plant medicine, especially the solubility, absorption, bioavailability. So these are the limiting factors for the herbal drugs, and thus when you formulate it into nanoparticles, we can modify the formulation characteristic. So when we convert them into nano size, it will help us to cross the barriers of increasing the time for prolonged circulation in the blood and making them fit as effective drug delivery systems. And green synthesis of nanoparticles is a very eco-friendly method, and it is cost-effective, can be used for large scale, and it doesn't have any use of temperature and toxic chemicals. So silver nanoparticles are an attractive smart structures which are having wide pharmacological activities, as you can see here, antifungal, anti-inflammatory, anti-tumor, antioxidative, etc. So coming to the characterization methods of silver nanoparticles include UV spectro analysis, FTR analysis, SEM and DLS, as well as XRA. So current status of nano-herbal medicine is a very good growing market, and by 2030, it will reach peaks. After extensive literature survey, we could find that boswellic acid is a potential lead molecule having anti-inflammatory activity and anti-cancer activity. So in order to repurpose it and to enhance the solubility and bioavailability of the boswellic acids to make it a targeted drug delivery system, we have formulated the boswellic acid silver nanoparticles. So boswellic acid silver nanoparticles, we need boswellic acid lead molecule from Boswellia serrata. We can obtain that. So the gum resin of Boswellia serrata was found to have high amount of boswellic acids, and the pharmacological implications of the boswellic acid include, it is very much useful in the chronic inflammatory diseases like rheumatoid arthritis, bronchial asthma, ulcerative colitis, and Crohn's disease. So it is very popular anti-inflammatory agent, and the novelty of our study lies in repurposing the boswellic acid as a COVID protein, COVID targeting molecule. So the rationale of work was framed to identify the boswellic acids and isolate them from gum resin of the plants, and then carry out the in silico docking on SARS-CoV proteins. So when you formulate the boswellic acid into nanoparticles, definitely we could see the improvement in the solubility, absorption, and also reduction in the dosage interval, which may ultimately reduce the dose-related side effects of the drug. So the present aim of the work was to synthesize the boswellic acid silver nanoparticles and to conduct the in silico docking studies along with ADMET studies on SARS-CoV proteins. The objectives were initially to extract and isolate the boswellic acid from the gum resin of the Boswellia serrata plant followed by carrying out the green synthesis of boswellic acid silver nanoparticles using concentration variation method, then characterize the boswellic acid silver nanoparticles using different analytical techniques like UV, SM, FTIR, DLS, and XRD. Then further to identify the dose ranging studies using acute toxicity studies of boswellic acid and its nanoparticles, followed by in silico docking and ADMET studies of boswellic acid on COVID proteins like 1S76, 6LU7, and 2HUZ. Further, the in vitro drug release studies were carried out on USB dissolution apparatus 1, followed by result and measurements and report of positive outcomes. So the plan of work was initially to collect the gum resin of the plant, authenticate, and then perform the oxalate extraction, identify the potential extract, and further carry out column chromatography to extract the boswellic acid, further green synthesize the boswellic acid silver nanoparticles using concentration variation method, followed by in vitro drug release studies, and then repurpose the drug by studying the in silico docking and ADMET studies. So overall methodology includes isolation of boswellic acid from gum resin of the plant, and then green synthesize taking lead as boswellic acid, and using concentration variation method, we could synthesize the boswellic acid silver nanoparticles and further characterize them, followed by in silico docking and ADMET studies, and ultimately studying the drug release kinetics. So this is the gum resin of the plant obtained from the Boswellia serrata plant. The concentration variation method for which green synthesis of boswellic acid silver nanoparticles was carried out. So the silver nitrate solution was prepared and 10 ml of the solubilized compound. That means 100 mg of the drug was taken in 10 ml and it was mixed with 90 ml of silver nitrate solution, kept for agitation for 24 hours on a magnetic steroid, and then centrifuged. The pale yellowish green color was converted into dark brown, which indicates the formation of formation of silver nanoparticles, and ultimately it was changed to brown. The extraction before the green synthesis, we could extract and isolate the gum resin, boswellic acid from the gum resin of the plant. So the gum resin was collected from the plant, and approximately 1000 grams was taken and loaded into the oxalate apparatus. Then the extraction was followed using N-hexane and methanol. Then the methanol extract was found to have high amounts of boswellic acid, which was column chromatographed to yield boswellic acid in pure form. The fraction of 70% ethyl acetate and hexane was discontinued, and 40 to 50% ethyl acetate and hexane mixtures could give pure boswellic acid fraction. This is the overview of the boswellic acid isolation and extraction, which was then identified by TLC as well as UV spectroscopy. And then that boswellic acid was utilized to convert them into nanoparticles. So the method what we followed could enhance the boswellic acid fractions, and we could get around 1.5% boswellic acid. This is a characterization of boswellic acids, and using UV spectroscopy, we could see the lambda max of boswellic acid is at 249 nanometers, and UV HPLC could give the chromatogram of boswellic acid. And coming to FTIR studies, we could see there is a strong OS stretching and ketone modification. So after conversion into the silver nanoparticles, the boswellic acids were identified using different spectroanalytical techniques, that is DLS, which could measure the particle size and zeta potential of the molecule. The particle size of the boswellic acid silver nanoparticles was found to be 21, which was 21.36, which was very much suitable for the targeted drug delivery. And from the literature, we could know that it may have an enhanced bioavailability. And the particles showed enhanced stability. By the zeta potential studies, we could measure a value of positive 24.15 millivolts, which indicates that it is having a good stability. The SEM studies also reported the particle size of 21 nanometers, and the XRD spectrum could relate that boswellic acid silver nanoparticles have a crystalline structure. The FTR studies could determine the formation of silver nanoparticles, where we can see the peaks near 700 centimeter inverse in the FTR, which indicate the formation of silver nanoparticles. Coming to acute toxicity studies, the experimental animals were taken, that is Wistar albino rats of 180 to 210 grams of either sex, and then those were fasted overnight prior to the drug administration, and they were divided into 6 groups and were administered with normal boswellic acid and the conventional boswellic acid as well as boswellic acid nanoparticles. And the observations of acute toxicity studies were according to OECD guidelines 423. They were initially observed for first 30 minutes and later for 24 hours and periodically for different timers. So any changes or the behavioral changes in the patterns were observed, and there were no changes observed. So the in silico docking of boswellic acid silver nanoparticles was performed on SARS-CoV proteins. So the 6LU7 protein is a protease functioning protein which can inhibit the viral replication. So boswellic acid has a strong interaction with the 6LU7 protein. That means it has a capability to inhibit the viral replication. Coming to 1S76 protein, which is a RNA polymerase dependent protein that also inhibits the viral replication. So the strong interaction of boswellic acid was also found on this protein. ACE2, coming to ACE2, which is a 6-MOJ protein, that ACE2 inhibitor acts as a carrier protein for binding of COVID proteins to the target receptor. So when this is inhibited, the target receptor can be inhibited and the boswellic acid could show a strong interaction with this protein. So we can say that boswellic acid, when it is repurposed as anti-COVID, definitely it can have a bright lead molecule structure. When the ADMET profile was found, it could show that it has strong solubility, protein binding, and enhanced bioavailability, but it could show endotoxicity. So when we convert the boswellic acid into nanoparticles, we can see reduced side effects, but the dosage is a problem. So that's why when we could perform the in vitro drug release studies using USP dissolution apparatus, we could see that there is a sustained release of the formulation, that is the converted boswellic acid silver nanoparticles rather than the conventional boswellic acid. So this is a novelty of the research study, where once we take the dose of the boswellic acid for one single dose, it can have a prolonged action. So that can reduce the dosage related side effects. So this is a drug release kinetic studies where we could prove that boswellic acid silver nanoparticles have sustained release nature. And coming to novel modification of boswellic acid nanoparticles, we could see 90% of the particles have around 21 nanometer size, and drug release kinetics could reveal that there is a sustained release nature of the boswellic acid, and in silico docking analysis and ADMET studies have proved the repurposed drug, that is boswellic acid silver nanoparticles, can be a lead molecule to the SARS-CoV proteins. So this can be formulated and developed as a product. Coming to future perspectives of the research study, still more size variation can be achieved, and in vivo in vitro correlation of the pharmacokinetic profile is needed. Furthermore, new pharmacological applications or new pharmacological activities can be explored. These are the references. And thank you.
Thank you, Dr. Sneha Thakur, for an excellent presentation. I see it's a good combination of lab work, and you have also implemented the docking studies for your repurposed molecule. And regarding the size of the nanoparticles, that is, it's around 21 nanometers, right?
Yes.
So that is that was determined by DLS.
Yes.
And you have also mentioned the zeta potential, which is quite good, which is less than -30. So it indicates the good stability of the nanoparticles. Have you also determined the PDI of the nanoparticles?
PDI, polydispersity index, no ma'am. We are still in process with that.
Okay.
Okay, that is yes, it's because when we are working with the nanotechnology or nanocarrier system, the stability is always issue, especially the long-term stability. So the size and the zeta potential and the PDI, these are the some of the important parameters that we have to evaluate at the time of manufacturing or a time of preparing, as well as a long-term stability also we have to check out, especially when working with herbal formulation. So, so I think really this is good work. You just check out about the stability of formulation, or especially the long-term aspects, maybe the 3-month or 6-month stability.
Yes, yes.
Yes, and one more point I noticed, you have also mentioned about the dissolution studies, right?
Yes, yes, ma'am.
So for nanoparticles, how did you proceed for the dissolution studies?
It
for
was
the silver nanoparticle?
Yes, ma'am. It was done
It was
using
like conventional or anything different, any modifications have you used for this method?
No, ma'am. We have used phosphate buccal, ma'am, as a medium, and we have taken that in USP dissolution. We have done using USP dissolution apparatus.
Okay, so nanoparticles as a dispersed material you have used, right?
We have packed in a muslin cloth and kept inside the basket.
Okay.
Dr. Sneha, you have used docking for your studies,
yes,
right?
Yes, yes.
So what was the docking score? You haven't mentioned.
Yes, ma'am. The docking score, the binding energy was -6 to 7.8 kilocalories, ma'am.
-6 to 7.8. Okay.
Yes.
Have you compared this with this standard drug?
No, ma'am.
You must compare this with standard drug. Then only you can support that boswellic acid can be the good lead.
Yes, ma'am. Yes, ma'am. Actually, from the
Both,
literature, we could know that this is the best
ma'am.
molecule.
Yeah, from the literature, there are so many molecules which are the best. Just like curcumin is also having the anti-inflammatory activities and anti-cancer activity. Then why not curcumin? Why boswellic acid? Because we have interest in the boswellic acid. That's why we are supporting for the boswellic acid, right, ma'am?
Yes, yes.
We must add binding energy and the docking score. If you are doing some docking studies, okay, which software you use for the docking?
Autodoc 4, ma'am.
Okay, Autodoc 4. And you must also add interaction diagrams so that we can see the amino acid proteins and the interaction, whether it is hydrogen bond, hydrophilic interactions, or van der Waals interactions.
Yeah, ma'am.
Okay.
It was hydrogen and hydrophobic interaction, ma'am. Both we could see in that figure.
Okay, it's okay. All the best, ma'am. Okay. Next presentation.
Thank you so much, ma'am. Thank you so much. Thank you so much, everyone, for giving me the opportunity.
Shall I invite next participant, madam?
Yes, ma'am.
Our next participant is Dr. Dinal V. Patel, presenting on topic formulations and evaluation of arginine-loaded solid lipid nanoparticles, affiliation, Assistant Professor, Department of Pharmaceutics, Faculty of Pharmacy, Dharmsingh Desai University, Nanded, Gujarat. Over to you, sir.
Okay, my screen is visible, sir.
Yes, madam, your screen is visible.
Okay, so good afternoon to all of you. My topic is the formulation and evaluation of arginine-loaded solid lipid nanoparticle. This is the content of my research work. So I'm starting with the introduction that there are a large variety of amino acids, or peptides, as well as fatty acid chains with various structure, length, and number, which explains their wide structural diversity and different physicochemical and biological properties. So, application of amino acid derivatives in different topical formulations were found in various marketed products, and from which the arginine containing internal as well as external products are very popular in the market. So these are some topical, I mean say external as well as internal products which contain the arginine, which is marketed. So arginine, that is basically the amino acid which is hydrophilic base, which is having the log K value that is minus 4.08. So that's why it is having very low permeability. Moreover, it is an amphipathic molecule, but due to the less hydrophobic portion, it cannot consider as the surface active agent. So there is a need to improve its permeability as well as its surface characteristic. As we see in the our biological cycles, this arginine will help in producing the cGMP, which causes the relaxation of muscles, as well as it produces the nitric oxide, which cause vasodilation and involve in opening of potassium channels. So this will supply the blood at the particular site of application, and in that way it will stimulate the hair. Moreover, metabolic pathway of the arginine results in ornithine, which converted into L-proline, and this L-proline is necessary for the synthesis of collagen. So collagen and other protein which are having very crucial role in the proliferation of cells as well as for the wound healing process. So for the different topical application, we can use the arginine molecule. The next material which I have used in my research purpose, that is the behnic acid derivative. It is having a 22 carbon chain, and one of the derivative of behnic acid, which is known as the glycerol behnate, or we can see the completol, that is very effective as the cosmetic ingredient due to its strong conditioning effect. So as compared to the esters of glycerin with other long chain fatty acids such as palmitic or stearic acid, the completol is having more pronounced hydrophobic property, which due to its longer fatty acid chain structure. So now move towards the our formulation, that is a solid lipid nanoparticles. So among the different nanoparticulate system, these solid lipid nanoparticles are made up of solid lipid core, which are stabilized by the surfactant molecule, and therefore it overcomes the limitation of associated with vesicle type of nanodrug delivery, which containing the liquid lipids such as low drug payload or carrier biotoxicity and physical stability, as in case of liposomes or myosomes. So the solid lipid nanoparticles are having number of advantages, which include that minor toxicity, then improving bioavailability. It prevents the degradation of incorporated molecule against the light, moisture, gases, and it will give the sustained release of the drug molecule. Hydrophilic as well as lipophilic API can be incorporated into the solid lipid nanoparticle. It can give prolonged drug release as compared to the nanostructural lipid carriers, and SLN can be produced by different type of techniques such as high pressure homogenization or solvent evaporation or ultrasonication method. So this is about my formulations. So now to develop this solid lipid nanoparticle, these four important matters are there. So first of all, the selection of drug. Drug may be either, we can use either hydrophilic or lipophilic drug. Then selection of lipids, then selection of surfactant which cover this solid lipid. And after doing the final formulation, we have to lyophilize that system. So selection of cryoprotector, that is also necessary for the stabilization purpose. So here, as I am incorporating the arginine into the solid lipid nanoparticle, so arginine, that is hydrophilic in nature. So very few studies are there which incorporate the hydrophilic drug into the solid lipid nanoparticle, because it is difficult to incorporate that hydrophilic molecule into the hydrophobic environment of solid lipid. So therefore, I am using in situ ion pairing concept to make my hydrophilic molecule somewhat lipophilic. So here I am using the in situ ion pairing agent during the development of formulation. So for that, I am choosing the oleic acid because it is biocompatible and it is having the higher log P value, that is 8. So it becomes a preferred candidate for ion pairing agent. So this in situ ion pairing concept is very simple and one-step process. The present study demonstrates the formation of in situ ion pair complex by change in solubility and partition coefficient value of the pure arginine. So the rationale of my project, that first one, that is the topical application of arginine with long chain fatty acid is beneficial for the overall skin health by moisturization. Then the hydrophilic nature of arginine limits its permeability through the topical drug delivery, and that's why I'm using in situ ion pairing approach to enhance the lipophilicity of arginine, which help in loading within the SLN. So SLN approach enhance the permeability of arginine within the skin layer for improving the skin condition. So these are the aim and objective. That my main aim is to develop the arginine loaded solid lipid nanoparticle employing the quality by design approach. So these are the material and method. Now first I am doing the estimation of arginine by UV spectrophotometry. So this is the calibration curve. This is the FTR spectra of arginine, which showing this main peak of the arginine molecule. DSC spectra of arginine is also confirmed that the arginine is there. So these are the preliminary, or we can say the physicochemical parameters of the arginine. So now the optimization of formulation by using QBD approach that involves first the quality target product profile, then identification of quality, critical quality attributes for the product, then identify the critical material attributes and critical process parameter using Ishikawa diagram and risk assessment study, and then formulation of SLN by using design of experiment approach. So these are the quality target product profile. So this will take the number of target product which optimize our product. Then these are the quality control attributes that I have taken the particle size and drug loading. This is the Ishikawa diagram, which involves all the factors which are environmental factors, machine factors, then material factors, which is known as the formulation factors, which affect these two critical quality attributes, that is the particle size and percentage drug loading. So this is the risk assessment by failure mode effect analysis method. So here the number of factors are chosen according to the risk priority number. So the factors which shows the highest risk priority number, they are taken for the further optimization study. So this is the preliminary study which characterize the arginine oleic acid ion pair. Here I am making the arginine oleic acid ion pair and evaluate it by performing the partition coefficient and solubility study. So here partition coefficient is higher for this ion pair, which conclude that the arginine molecule is converted towards the lipophilic nature. then the solubility in water is decreased, which also confirms the formation of arginine oleic acid ion pair. So this is the formulation method for arginine loaded solid lipid nanoparticles. Here, compritol and oleic acid that is melted at 75 degrees, as well as water and arginine that will also heated at 75 degrees Celsius, and this both solution are homogenized in the ultrathorax. So meanwhile, water and poloxamer, here we are using poloxamer as the surfactant molecule. So water and poloxamer is also added into this mixture into the ultrathorax. So after homogenizing, that emulsion is known as the pre-emulsion. And when we put this pre-emulsion into the probe sonicator, then it will formalize as the final solid lipid nanoparticles, which are centrifuged at 1000 rpm for 30 minutes. And that solid part is taken for the further study for XRD or DS-like studies. So these are the preliminary batches which I have prepared for the solid lipid nanoparticles, in which I have taken this number of factors. Okay, so from this study, we have concluded that if we combine these two surfactants, that is poloxamer 188 and 280, then it will give the lower particle size and somewhat higher drug load, and concentration of surfactant. If we use it within 2 to 5%, then it will affect the particle size and drug loading. Another thing that the concentration of oleic acid, which is in the range of 0.5 to 2%, that will also affect the drug loading. So by using this preliminary study result, we will take the, I'm taking the some factors for design of experiment. So this is just one of the particle size determination for this first trial batch, which is having 1376 nanometer particle size. So I'm using after then the 2⁵ full factorial design, in which I have taken x1, x2, and x3 factor as the concentration of poloxamer, then concentration of oleic acid, and sonication amplitude. Because this x1 and x3 factor having the major effect for the particle size, and due to using this oleic acid as the in situ ion pairing agent, the drug loading will be increased. So I am taking these three factors in the study, and as the response, particle size and drug loading was taken. So this is the formulation of 8 batches according to the design of experiment software. Now I am doing the evaluation parameters for all the batches, that is encapsulation efficiency and drug loading capacity, then particle size distribution and morphology, and in vitro drug diffusion studies was also performed. And after evaluation of these all batches, I have optimized one of the batch for which I have performed the ex vivo drug permeation study and retention study, DSC, XRD study, FTIR, time analysis, and stability study. So these are the observed response for the 8 experimental batches, in which the highest drug loading will be observed for this 7th batch, and the lowest particle size will be observed for this 8th batch. The PDI also within the range of is approximately 0.5. So these are the data analysis for the design of experiment. This is the ANOVA result, which suggests the 2FI model, that is 2-factorial interaction model. These are the 3D response plot for the particle size, which clearly shows that if we increase the sonication amplitude, then particle size will be decreased. And similarly, the effect were shown. This is the counter plot and 3D plot for the percentage drug loading. These are the constraints which I have taken, from which I have chosen the 3 checkpoint batches. This is the optimized formula, optimized batch, and these are the 3 checkpoint batch for validation of design expert software. So these are the evaluation parameter for the optimized batch, which shows 273 nanometer size.
Dinal, I think time is over, so you try to conclude your presentation.
Okay, okay. So these are the in vitro drug diffusion study for 24 hours, and it follows the zero order and phylogenetics. This is the drug permeation study in which the more percentage of cumulative amount of arginine will be seen in case of a solid lipid nanoparticle based cream as compared to the conventional cream. This is the drug retention study on the skin layers. So on the skin layers also, after 24 hours, SLN based cream shows the highest accumulation. So these are the IR spectra for the SLN and pure arginine, which shows the amorphous peak. Same DSC spectra for the pure arginine, and this black that is for the SLN. These are the XRD spectra, which shows the for SLN and pure arginine. This is the TAM analysis for the optimized batch, and stability study will be performed for 6 months according to the ICH guideline at 40 degrees Celsius and 25 degrees Celsius. And these 3 parameters were checked, which are having very less changes during the stability study. So the present research revealed the significance of QBD approach in developing the novel drug delivery system, and by utilization of all the concepts, the SLN containing concentration of holoxamer, which is having 2%, oleic acid 1.5%, and sonication amplitude of 59.78% was found to be optimized one, which are having the particle size of 273.6 nanometer and 8.57% of drug load. So this I have published in JDDST journal, same article. So thank you.
Thank you very much, Dr. Dinal Patel, for a very nice presentation.
Thank you, ma'am.
I have a question about the permeability studies. I don't know whether I missed. Have you mentioned what membrane have you used, whether it is skin, like or animal skin or animal
yes,
skin?
ma'am. I have used the Wister albino rat for that, and I have also taken the permission for from Animal Ethical Committee for that. So I have used the rat skin for the study in France diffusion cell.
Okay. And did you treat the skin before doing the studies, or like?
Yes.
What?
Yes, ma'am. I have properly clean that skin and keep it in the ethanol also to remove the dust and all the things before the study.
Okay. And you mentioned that these solid lipid nanoparticles will be effective for the skin conditions.
Yes, yes.
Right? Because you are basically increasing the hydrophobicity of one of the very, very hydrophilic amino acid, right? Arginine.
Yes.
So do you have any specific skin conditions that this can be used for, that you recommend?
Yes.
What is the best condition that is useful?
Ma'am, still the marketed preparations have used it to increase for the libido, for the treatment of libido, and for the hair shampoos also. But this can be also utilized for wound healing purpose also. Wound healing, that is also the application for the arginine, topical arginine preparation.
Okay. Thank you very much for a nice presentation. And if any other chairperson have question, please proceed.
Dr. Dinal, I have one question, very simple question that so many research is going on the nanoparticles. So how your research is different from the others? What is novelty in your research?
Yes, ma'am. When I started my work, ma'am, then I'm taking the green, green formulation for that. So amino acids are the molecule which are having no any side effect and which are incorporated within our body also. So if we use such type of nutrition as a drug, then it is beneficial as compared to the other synthetic.
Okay. Okay. All the best.
Thank you.
Shall I call the next participant, madam?
Yes, ma'am.
Please. Yes. Madam, can you please stop screen sharing? Now I would like to invite Mr. M. Mohammad Sahin Shah, presenting on topic pharmacognostical, phytochemical, antioxidant and cytotoxic evaluation of Rhynchoglossyum notoneum. B.L. Burd, Unutilized Nature's Treasure. Affiliation, Research Scholar, Department of Pharmaceutical Technology, University College of Engineering, Anna University, BIT Campus, Tiruchirappalli, Tamil Nadu. Over to you, sir.
Hello, good afternoon, everyone.
Good afternoon, sir.
Was disabled for.
Yes, I have.
I'm not able to. Yeah, I'm not able to share my screen.
I made you the host, sir.
Yeah, my screen is visible, ma'am.
Not visible.
Try to share it again.
In my screen, it is mentioned host disabled participant screen sharing.
Okay, 1 second, wait. Host Institute, please. Okay, yeah, now you can.
Okay. Yeah, good afternoon, everyone. I am Mohammad Shaheen Shah, research scholar from Department of Pharmaceutical Technology, Anna University, Trichy. And my topic is pharmacognostical, phytochemical, antioxidant, and cytotoxic evaluation of Rhynchoglossyum notoneum, which is an unutilized nature's treasure. The macroscopic, microscopic, phytochemical, and physiochemical properties of Rhynchoglossyum notoneum and total phenolics, total tannins, total flavonoids, proanthocyanidin, beta carotene, and lycopene content were quantified in this research, which revealed that confirmed the presence of significant phytoconstituents. The antioxidant potential were determined by 3 different methods, and antimitotic activity screening was done using the novel mung bean method, mung bean antimitotic assay, and the cancer chemo preventive activity of the plant were documented for the first time as a potential source for discovery of novel drugs. Sri Lankan government introduced one postal stamp for this underutilized medicinal property, ethnomedical importance plant. Rhincoglassum rotanianum is a succulent herb and a native of Asia. Its synonym is East Indian Kulugia in English and Neer Sambarani in Tamil. These species, few species are found in India, and ethnomedical information revealed the usage of the treatment in polio and inflammation by the rural tribes of Nilgiri district, Tamil Nadu. This is the plant image. The objective of the study is macroscopic and microscopic study of Rhincoglassum rotanianum, and to perform phytochemical screening and determination of physiochemical parameters and the antioxidant potential of the plant, and to study the antimitotic and the chemopreventive activity of the plant. The significance of the study is macroscopic and microscopic study is useful in identification, authentication, and preparation of monograph. Quantification of secondary metabolites and antioxidant activity confirms the presence of a significant amount of phytoconstituents, and antimitotic activity screening using the green-gram model revealed the significant inhibition of antimitotic activity of the plant, and in vitro chemopreventive activity using HeLa cell lines reveals the tremendous anti-cancer activity of the plant. The methodology part, these methods are used for qualitative and quantitative methods. Pharmacological activity screening was done. Antioxidant activity by DPBS radical scavenging, peroxide ion scavenging, and nitric oxide scavenging activity. The reference are mentioned in front of each respectively, and antimitotic assay by Kumar Singhal method, MPT assay, the method described by Mosman. The results of the study is the macroscopy of the plant reveals the leaves are alternate and unequal of cordate. Texture of the leaf is very thin and delicate. Lamina is obliquely ovate and cordate. Leaves are very shiny and dark in color. The microscopic character of the microscopic character are shown here. The petiole, stem, and TS of the leaf through midrib. The single layer of epidermis and 3 to 4 layer of hypodermis. Then vascular bundles are present in the stem. The powder microscopy showed these characters: xylem vessels with spiral thickenings and horizontal slits, xylem fibers which are longer than xylem vessel but thinner in appearance. It occurs as a multiple strands. The stomata is cruciferous stomata, and mesophyll tissue found in bunches of varying sizes. There are two types of trichomes are present: glandular and non-glandular trichomes. Angular shaped pyramid-like trichomes with one or two cells. Phloem fibers of lignified and non-lignified in nature. The quantitative microscopy revealed the stomatal number in upper epidermis and lower epidermis was found to be 6.6 and 83.9 respectively, and stomatal index in upper and lower epidermis is 18.26 and 29.17. Vein islet number was found to be 2.5. Vein termination number is 2.3 and palisade ratio is 2.5. The physical parameters were determined. The total ash value, total water soluble as, acid insoluble as, volatile oil content, and foreign organic matter loss on drying foaming index were determined. Volatile oil content is nil and the total ash is 14.205%, and water soluble as is 7.167%. Acid insoluble as 0.149%. Foreign organic matter is 0.41%. Loss on drying is 9.085%. Foaming index is less than 100. The extractive values showed that 70% of ethanol extract gives a maximum extractive value of 17.21% next to ethanol extractive value, that is 14.484%. Phytochemical screening using the various extracts were done. The 70% ethanolic extract shown to have phytosterols, carbohydrates, proteins, tannins, glycosides, mucilages, and flavonoids. The quantification of secondary metabolites were done, and the total phenolic content was found to be 109.46 milligram, and total tannins to 249.5, total flavonoids 116.28, proanthocyanidin 158.58, beta-carotene 1.729, lycopene 0.182 microgram per ml. Antioxidant activity screening was done with the DPBH radical scavenging, peroxide ion scavenging, nitric oxide scavenging activity using ascorbic acid as a standard drug. Rhingoglassum nothanianum gave the result of IC50 value of 93.51 microgram per ml in DPBH radical scavenging activity, whereas ascorbic acid shown to have 18.04 microgram per ml IC50 value. Peroxide ion scavenging, same like IC50 is 81.34 for the plant and 92.42 for the standard ascorbic acid. Nitric oxide scavenging activity give the IC50 value of 14.19 microgram per ml, whereas ascorbic acid shown to have found to have 81.96 microgram per ml. Green gram antimitotic assay using the mung bean. We use the green gram Vigna radiate, it's called as mung bean. The water imbibition at a higher concentration completely inhibited the weight gain, thereby seed germination and the growth retarding effect, and radical decay was resulted. The growth retardation with our plant could have resulted from the inhibition of mitotic cell division and radical protrusion brought about by the osmotic stress. The present study, the growth retardation brought about by the Rhingoglassum nothanianum was found to be associated with a significant reduction in mitotic index. The inhibition, whether the inhibition is specific for cancer or is incidental, was determined by cross-checking the inhibitory activity of anti-cancer drug, standard anti-cancer drug, cisplatin. Here you can see the controlled tape water treated mung bean imbibed well, and the germination started. The radical length also good. Where the cisplatin treated group, even from 0.5 mg, we took 5 different concentration. There is no radical growth, no inhibition in 2.5 mg per ml. There is no radical growth, no inhibition, same like. And our plant shown the dose dependent reduced inhibition, reduced radical length, and reduced germination. This graph clearly shows the reduction, dose-related reduction in water inhibition of green gram seeds by our plant Rincoglasum nothanianum and by standard drug cisplatin. The effect of radical length of green gram by the drug and the effect on germination of green gram seed was shown in this graph. The MTT assay was carried out to know the chemopreventive activity of cancer by the plant. Rincoglasum nothanianum was evaluated for its cytotoxicity against HeLa cell lines in different concentration, 31.25, 62.5, 125, 250, and 500 microgram. The IC50 was found to be 124 microgram per ml, and it clearly shows the tremendous anti-cancer activity on cervical cancer. The IC50 value calculation done by this graph, IC50 is 124 microgram per ml. Study highlight is the investigation results were documented for the first time, as there are no scientific documentation on this plant in pharmacognostically or pharmacologically anywhere. And significant discoveries, unutilized nature's gift. This plant, Rincoglasum nothanianum, could be the potential source for discovery of novel drugs. Elaborate research has to be carried out to explore medicinal usage of underutilized and unutilized medicinally important plants. This is the thing that we need urgently nowadays. The plant under study contains a significant amount of promising pharmacologically active phytoconstituents. Its antioxidant activity clearly be the base of anti-inflammatory, anti-arthritic, antimicrobial, hepatoprotective, and anti-cancer activity. The areas of improvement, the elaborate studies have to be conducted on the isolation, identification, characterization, and the pharmacological in vitro and in vivo screening. The high throughput screening revealed this plant has a good anti-cancer activity and antioxidant activity. The pharmacologically potent moiety from the elaborate studies can be used as a lead compound for designing a potent anti-inflammatory drug, which can be used for the treatment of inflammation and inflammation-mediated diseases like cancer, neurological disorders, and even in aging also it can be used. Thank you everyone.
Okay, good presentation. One thing, is there any formulations available in the market of this particular plant?
No, sir. This is the this is ethnobotanically the medicinal property was documented by one journal, but there is no studies available on this plant still.
Even even formulations are also not available.
Yeah, not available, sir. But the irregular tribes from Nilgiri district of Tamil Nadu, India, they are using for using as an anti-inflammatory, and even they are using as using for polio.
Okay, so there are the traditional uses are there.
Yeah, traditional uses are there, sir.
Okay. Any questions from other chairpersons? Okay. Yeah. Thank you very much.
Thank you, sir. Thank you.
Yeah. Thank you very much.
Thanks, everyone, sir.
Shall I call the next participant? Madam?
Yeah, sure.
Sir, our next participant is Mr. Monu Kumar Shukla, presenting on topic development of fluorescein conjugated carbon nanotubes of methotrexate with a chemo enhancer and a stealth agent for the diagnosis of anti-cancer activity in A549 cells. Affiliation, research scholar, Department of Pharmaceutical Chemistry, School of Pharmaceutical Sciences, Shulini University, Solan, Himachal Pradesh. Over to you, sir.
Good evening. Is my slides are visible?
Yes, visible.
Yeah. Thank you so much, Association of Pharmacy and Pharmacy Professionals and St. Paul's College of Pharmacy for providing us opportunity to present our research work on this conference. Good evening to all of you. Myself, Monu Kumar Shukla, and my research topic is development of fluorescein conjugated carbon nanotubes (CNTs) of methotrexate with a chemo enhancer and a stealth agent for the diagnosis and anti-cancer activity in A549 cells. So the overall work is going to elaborate about the on-off mechanism of fluorescein conjugated carbon nanotubes for real-time drug release. Carbon nanotubes are advanced nanocarrier for the delivery of anti-cancer drugs. In the field of carbon nanotubes, there are both single-walled carbon nanotubes and multi-walled carbon nanotubes. So both are used for targeting delivery of anti-cancer drugs where the high therapeutic effect is necessary. So there are several regions to choose carbon nanotubes for as a model delivery systems. Which are they? The first one is they are versatile platform that can be used to attach a wide range of chemical moieties. And the second is their backbone is not degraded intracellularly, while the attached molecules are released by cleavage of covalent linkages with other compounds. And the third one is they can be easily separated from the released drug. So in our study, we used methotrexate, which is already reported or approved drug for anti-neoplastic or anti-cancer drugs and they are reported for multiple types of cancers. So MET is chemically stable and contains carboxylic groups, which could be easily esterified with fluorescein. and so that this advantage of MAT may make it a model compound for our study. So the NDDS, the novel drug delivery systems like nanoparticles pose another challenge. What's that? That is accumulation of drug in organs or tissues by the mechanism of RES, reticuloendothelial system. And the organs involved in RES system are liver, lungs, and spleen. So due to this mechanism of RES system, so drug concentration in the targeted site is decreases, and therapeutic activities also decreases. Whereas the drug is accumulated in the other organs due to RES system, so the drug concentration will be increased in non-target sites like RES systems organs, and due to this it will also increase the toxicity. So many studies are reported that the dextran sulfate DS is helpful as a stealth ligand in evasion of RES system. That means it will inhibit the RES system, as well as increases the cellular uptake of multi-wall carbon nanotubes. So in our studies, we used multi-walled carbon nanotubes. So this will also reduce the accumulation of drug in organs and increases the availability of drug at the target site. So that's why we are also using DS, dextran sulfate. On the other hand, so what is chemo enhancer? Chemo enhancer are compounds which increases the anti-cancer activity of other chemotherapeutic or anti-cancer drugs. So in our study, we used low dose of suramine, which is a reported antiparasitic or reverse transcriptase inhibitor, which is already reported. And we used this drug as a chemo enhancer with our methotrexate. So how this drug is working? It inhibits the multiple pathways like fibroblast growth factor, epidermal growth factor, WNTG, and protein and STAT5 signaling. So in our study, we developed a system that can be used as platform technology for real-time drug release monitoring. We developed a carbon nanotubes based system to determine the release rate of covalently linked drug, that is methotrexate. Additionally, the possibility of employing a chemo enhancer, that is suramine, and a stealth agent, dextran sulfate, has been explored. So what is the basic hypothesis behind this research work? So real-time detection of drug release by the conventional pharmacokinetic approaches is confounded by low sensitivity. In the case of tissue targeting of nano drug delivery systems, where the low concentration of the drug is reaching in the systemic circulations. So we present a novel fluorescent sense turn on platform for the real-time monitoring of drug release from the nanoparticles based on reversible fluorescence quenching in fluorescein esters. So dynamic fluorescence quenching in fluorescence esters allow us to monitor drug release from the nanoparticles in real time. And the fluorescence is quenched due to the covalent link of drug molecules from the drug molecule to the fluorophore via ester bonds, that is FITC and with multi-walled carbon nanotubes. So hydrolysis of ester. So hydrolysis of ester bonds by the action of intracellular esterase results in activation of fluorescence, and the enhance in the fluorescence intensity correlates the amount of drug release. Before going to the further slides, I want to introduce our system first so that we can understand easily in the next slides. So what we develop here in the picture in the left side, multi-walled carbon nanotubes is linked with FITC. And this FITC is attached with methotrexate via ester bond here. Okay, so when we go for the hydrolysis, so at the time of hydrolysis, the methotrexate drug will detach from the FTC and it will produce some fluorescence. So we can identify that the drug is released. If the fluorescence is not produced, so we can say that methotrexate does not detach from the FTC. Okay, thank you so much. And so we are going for the next slides. So this slides belongs to the preparation and characterization of fluorescein labeled multi-walled carbon nanotubes. So amino-functionalized multi-walled carbon nanotubes and FTC were first stirred with overnight with multi-walled carbon nanotubes and FTC were stirred overnight in 10 ml of carbonate buffer having the pH of 9, and then this conjugate was separated by the filtration and washed several times with water and acetone to remove the unreacted dye. The conjugation was confirmed by the formation of fluorescence at 488 excitation and 530 nanometer at emission. So the yield was of FTC attached multi-walled carbon nanotube was more than 95%. And we prepare here in our research 4 types of formulations or conjugate system. The first one is FTC attached multi-walled carbon nanotubes. Second one is MET conjugated FTC multi-walled carbon nanotubes, and third one is suramine attached methotrexate FTC multi-walled carbon nanotubes, and the fourth one is dextran sulfate methotrexate FTC attached multi-walled carbon nanotubes. So after the preparation of FTC multi-walled carbon nanotubes, they were incubated with methotrexate for the 24 hours. The same protocol was followed for the suramine conjugated and dextran sulfate conjugated multi-walled carbon nanotubes. So these 4 preparations were done in our research. So in the figure, you can see the fluorescence was on when it will hydrolyzed, and off it will not hydrolyzed. So this is also so. We also prepare 2 types of formulation, and when hydrolysis was done, so it was dissolved in the water, and for storing our conjugate system, we also dried for that. So whenever we use, we just dissolve or disperse in the water. So so this is in the left side, the image is TAM of a methotrexate FTC attached multi-walled carbon nanotubes. The hydrolysis profile of methotrexate FTC conjugated multi-walled carbon nanotubes. So we have 2 figures. The A is presented hydrolysis profile of methotrexate FTC multi-walled carbon nanotubes in phosphate buffer saline having the pH of 7.4 and A549 cell lysate. Whereas in the B second graph, a correlation between the drug release and fluorescence intensity in the presence of A549 cell lysate. Hydrolysis and cytotoxicity profile of methotrexate conjugated FTC multi-walled carbon nanotubes. So in the table 1, we are presenting here the hydrolysis of conjugates in phosphate buffer saline A5 with A549 cell lysate and A549 cells. So for this, we use 3 types of conjugate systems. First one is methotrexate conjugated. Second one is suramine conjugated with methotrexate, and third one is methotrexate and DTAF. That is also we prepared earlier. So we used for this hydrolysis. So here And the second table belongs to the cytotoxicity of conjugates in cell lines. A549 cells, MCF cells, and MOLT4 cells. So here we used different type of conjugate system. First one is free methotrexate. We treated all three cells via free MAT, methotrexate, and methotrexate conjugated FITC multi-walled carbon nanotubes, suramine methotrexate conjugate system, methotrexate and DTAF conjugate system, suramine methotrexate and DTAF conjugate systems. Cytotoxicity profile of free methotrexate and drug conjugates. So the first graph belongs to the percentage viability between percentage viability and methotrexate concentration, where first A and B is FITC conjugated multi-walled carbon nanotubes in A549 cells. Effect of conjugate system on A549 cells. So we also check the effect of conjugate systems on cellular and molecular level of A549 cells with controlled on the basis of DNA damage, P53 levels, and PCNA levels. So we treated we work with a control and FITC conjugated multiple carbon nanotubes and methotrexate conjugated systems, suramine methotrexate conjugated system, and free methotrexate. We also we actually work these systems for 72 hours, and the DNA damage were determined by the diphenyl assays, amine assays, and P53 PSNA levels by whole cell ELISA. Quantitative and relative cellular uptake of FITC multi-walled carbon nanotubes and methotrexate conjugated FITC multi-walled carbon nanotubes. So in the first graph, quantitative cellular uptake of both conjugated system in the presence of 100 micromolar MET methotrexate and 100 micromolar folic acid at 4 degrees Celsius was estimated. And second graph is the relative cellular uptake of cell lines expressed in the ratio of cellular uptake of methotrexate conjugated FITC multi-walled and FITC multi-walled conjugated systems. So quantity, then we also done quantitative cellular uptake of methotrexate FITC multi-walled conjugate nanosystems and dextran sulfate conjugated methotrexate FITC conjugated systems. So here we used A549 cells and draw 64.7 cells. For this, we incubated the cells with these both conjugated systems for 6 hours, and cellular uptake was determined. Cellular uptake of dextran sulfate and suramine with conjugated with methotrexate FITC multi-walled carbon nanotubes. So in the presence of this, we also used different inhibitors for active uptake, like 4 degree. We check the cellular quantitative cellular uptake of these systems with 4 degrees Celsius, sodium HID, chlorpromazine, hydroxypropyl, beta cyclodextrin. So where chlorpromazine shows the more cellular uptake as compared to other systems, solvents. So what was the conclusion? We concluded, we reported a novel fluorescence turn-on theranostic platform based on multi-walled carbon nanotubes in which the fluorescence is quenched due to the covalent linkage of drug molecules to the fluorophore, that is FITC, via ester bonds. The hydrolysis of ester bonds by the action of intracellular esterase result in activation of fluorescence, which ensure the increment of fluorescence intensity correlates with the amount of drug release. And supramolecular functionalized with suramine could increase the activity of anti-cancer compounds, while dextran sulfate could evade the RES systems, thereby suggesting their potential application as a chemo enhancer and stealth agent respectively. Although we also conjugated these conjugate systems have been employed as model nanoparticles and methotrexate as a model in the present study. The general chemistry reported here could be extended to other nanoparticles and anti-cancer drug molecules. So what are our future work plan? We are planning to optimize our formulations, our formulation of multi-walled carbon nanotubes for other in vitro and in vivo activities. And we are also planning for docking and molecular simulation studies. So Dr. Deepak Kumar is my supervisor. He is an associate professor in Department of Pharmaceutical Chemistry, School of Pharmaceutical Sciences, Seoul University. So these are my lab mates. Thank you so much.
Thank you very much, Mr. Monu Kumar Shukla, for a nice presentation. It's a little bit unconventional, what we can say, because less number of people work with the carbon nanotubes. In India, especially, I don't see much research work on the carbon nanotubes. I have a question about the preparation of the multi-walled carbon nanotube that you mentioned, because in the starting material of your formulation, you have used the amino functionalized a multi-walled carbon nanotube. So before that, you developed it in the lab, right?
Yes, ma'am.
Multi-walled. So can you please show that?
We actually it was a gift sample from other labs. So we asked for the gift sample. So actually we done the rest of the work. So we didn't prepare the multi-walled carbon nanotubes.
Okay, that's what I wanted to know, and that's why I see that that method was not included in that. Okay, so you got it from any Indian company or outside India?
Actually, one of my my supervisor's friend from South Korea, he sent as a gift sample.
Okay, and then you use that as a starting material to synthesize your next molecule. Okay. So the target of this delivery system is to develop it as a theranostic, right?
Yes.
Okay. And what is the purpose of that particular cell that you have used?
A549 cells.
Yeah.
It's a cell belonging to the lung cancer. And the purpose of this overall research work was to check the delivery system, actually monitor the real-time release of the drug from our systems. So we attached our anti-cancer drug with FITC, and with we develop a ester bond. Whenever the hydrolysis will take place, so the drug will detach from the FITC. And first one is multi-walled carbon nanotubes, which is where FITC is attached. And this FITC is attached with the methotrexate via ester bond. Whenever the ester bond will break via the hydrolysis, the fluorescence will produce.
Okay.
And if the hydrolysis, if the ester bond is not breaking, so the fluorescence will not produce. So we can say that how much the drug will release from the systems.
Okay.
Yes.
And how this dextran sulfate is used as a stealth agent, right?
Yes.
You mentioned it as a. So on what basis do you claim? Based on literature or based on your own studies that it is a stealth agent?
Yes, ma'am. Actually, it's not a new. thing, and it was already reported. It was reported for the magnetic nanoparticles, where dextran was used as a stealth agent. So why we are using dextran here, dextran sulfate? Actually, whenever we are going to prepare nanoparticles, we always facing an accumulations via RES systems, reticuloendothelial systems. If the RES systems is every time it will cause some problems whenever we are using nanoparticles. So whenever you using nanoparticles, due to the RES system, the nanoparticles will accumulate in the liver and other organs. So here we are preparing nanoparticles of multi-walled carbon nanotubes. If we are using dextran sulfate, so it will reduce or inhibit the RES systems. So if we are using 1 mg of drug, so it will not accumulate in the liver or other organs. It will be available at the target site.
Basically, it will escape the RES, right?
Yes, yes.
And it will enhance the circulation time, or what you can say is the longevity of the nanoparticles, right?
Bioavailability of the drug.
Yes.
Yes.
Okay. Best wishes for such a nice presentation, and thank you for your enthusiasm and sharing this knowledge with the audience. Thank you.
Thank you so much.
Over to you, Dr. Mahul.
Yeah. Okay. I think it's really because, as ma'am told, that the carbon nanotubes, and this is one of the new era. Generally, it is we have seen lots of work, especially in the surface chemistry, and we have also here surface science and nanotechnology centers where they are working. Okay, so is there any applications into targeted drug delivery, or that we can utilize such kind of system?
Yes,
for
sir,
drug
definitely,
targeting?
Sir, you know, for example, if we are targeting a colon, so we can use folic acid as a targeting systems. So if we are using methotrexate to treat colon cancer, so we have to attach folic acid in these systems. So there are folate receptors which is highly expressive in nature in the condition of colon cancer. So we can target colon cancer by attaching folic acid in this system.
Okay. Yeah.
Yes.
Mr. Monu, your work is really very rare and interesting. I think you must file a patent after doing your research work. It's going to be very beneficial for the society too. All the best. All the best.
Thank you.
Okay. Yeah.
Yeah. Thank you so much, APP and St. Paul College of Pharmacy. This is my second conference. The first was in 2020 in January, where I was worked in CSIR IIM Jammu for 3 years. And recently I joined Suli University as a PhD JRF under Dr. Deepak Kumar. Thank you so much for giving me opportunity to present our work in this prestigious conference. Thank you. Thank you so much.
So where you presented the first time, what you are saying?
Sir, I also present my work in 2020 at CSIR IIM Jammu, and the conference was with one of the pharmacy college in Ahmedabad.
In Bhopal or Ahmedabad?
Ahmedabad, sir.
Okay.
Gujarat.
Okay.
Yes, sir.
So, okay, thank you.
Thank you so much, sir. Thank you, sir.
So, thank you all the oral presentees. All presentations were very informative. And I also wish to thank all the judges for their valuable time, and the discussion happened. I think it will be useful for the research project of our students. And if any judge or chairperson want to share any view, or otherwise I can switch over to the anchor, please. Thank you.
Yes, I have something to say about all the oral presentations, like, because as you know that all of you are competing here for the best oration, and it is also related with some prize, but I really appreciate the efforts of you people to participate and sharing your research with us. So it was really nice experience for me to involve in your research work and to look at all the data, how you are doing, how you are presenting, and what are your thoughts about that. And I hope that if whoever the students who are pursuing the degrees like MPharm or PhD projects, they must be benefited from what you have shared here. So I hope for all of you, it is a good experience, and so as well for us also who are being here as chairperson. So I really appreciate the efforts, and I hope to have more participation in the future conferences. And again, many thanks for to the organizations. I know they have put a lot of efforts to make this conference, and I think first day earlier it was planned to be by physical or hybrid mode, one day virtual, one day this, but because of the current pandemic condition, you have been shifted to both day virtual. But anyhow, it's a very one of the what you can say that a very memorable conference for me. Thank you very much.
Thank you so much, ma'am. Thank you, sir.
It's over to anchor.
Mr. Monu sir was the last participant. I would like to congratulate all the participants who presented today, and I would like to thank all the judges, everyone who gave us your precious time. Thank you very much.
What's your name, madam? We have not seen.
I am Suhaira Sahi, sir.
You are faculty there or student?
Yes, sir. I'm faculty here.
Okay, good. So we conclude here. Thank you, Dr. Kiranmai, madam. So in the morning, I think first session will be at 10. So what is the time?
Yes,
so
sir.
we will join tomorrow morning. So it's good morning here. So your is good evening.
Yes, sir.
So now
We can understand. So it's very difficult,
it is 7
sir,
a.m.
for
7
you
a.m.
people, really. It's very difficult for you people to stay like the same enthusiasm you are showing. It's a very good, we should learn this patience from users.
So it was actually our night show, 12 a.m. to 7 a.m.
Yes, sir. See you, sir, then tomorrow.
So, but it was a combined effort. So,
yes.
Thank you all for your effort. So, see you tomorrow then.
Yeah.
Yes.
Thank you.
Okay.
Good evening,
bye.
all.